Good morning, everyone. Okay, sorry. I try to speak without microphone, and so it's a pleasure to see so many friends, and I hope that we will have many discussion about biology, our single cell spatial transcriptomics, and everything that we will discuss in these meetings. I'm sure that there are many of you that are new to this meeting, and I really thank you for coming here. And you will see that this community is going to welcome you and in many discussion and possible collaboration. So please interact with everyone here. So let's see if we can get the slides going. And I have mostly housekeeping announcements. So first of all, so you have welcome to the Human Cell Data General Meetings. You can have all the information here at the website, and follow us on Twitter. And if you wish, please add your comments at this hashtag. Add your comments so the whole community and your colleagues will be updated, and will also learn about what we are talking here at the meeting. This does not move. Okay. Okay. Then I need to be stereo, right? Okay. No, just. So one notice. So we are going to record, and we are going to broadcast live, and we may use the material at this meeting for our to further advertise the project. So if you don't agree to be in pictures or be in the video, please be aware. And if I'm sure that all of you will be okay probably, but yeah, just be aware. If you don't agree, so please be aware. And you probably you shouldn't be in this room if but I'm quite sure that we will not have any problem for this. Also very important to thank all the sponsors, and in particular I'd like to thank the Helmsley Charitable Trust for their foundation level support for the 2024 Human Cell Data General Meeting. And also, I would like to thank also 10x Genomics, also for their foundational level support for the 2024 Human Cell Data General Meetings. And as always, please visit the sponsor. The sponsor make this meeting possible, so please spend their time. Also, because the sponsor have many interesting projects that will help our research. And additional sponsors are here, are those bronze level sponsor: BioLegend, BioScript Genomics, Cypher, Curio Bioscience, Euroclone, or Euroclone in Italian, and Scale Bioscience and Visgen. And also our exhibitors: Element Bioscience, Illumina, Takara, Oxford Nanopore Technology, 10x Genomics, Ultima Genomics, Visgen, Scale Bioscience, and Pars Bioscience. And also for our support, for our operation, we have support from Chen Zuckerberg Initiative, the Klarman Family Foundation, and the Wellcome Trust. Thank you for all of you for supporting us. Some more housekeeping. So for the safety, there are two safety officers. One is Christine, that is there, and probably all of you will know already or by the end of the meeting. And the other is myself. Any concern that you may have, please do not hesitate to contact us. Also, please always wear your name badge. This is always very important for us to understand who is that only us is at the meeting. It's very important to understand who is also for the distribution of the meals. And also for the COVID-19, if you suspect that you may have COVID, or please refrain, or perhaps any other call, please wear a mask like Aviv is doing, and she doesn't have COVID, okay, we talked before, okay? And just a common cold, but I think is definitely is a good practice. And whatever you know, if you have any additional question, please visit our registration desk or mail us at the Human Cell Atlas. We have a code of conduct here, and which aims at you know. No, it is very long, and I'm not going to read everything. But essentially, what we would like to have a good discussion with a very good spirit, a constructive spirit, and we do not want to. We can have a discussion, but always in the proper. With the proper manner. I'm gonna read here about harassment. So we are really. We really want to prevent any sort of harassment. So harassment in speech, behavior or action that is unwelcome or offensive to another person, whether based on sexual orientation, gender identity, race, ethnicity, culture, social and economic class, education level, sex, age, political beliefs, religion, military status, or physical or mental ability. Okay, so please keep in mind always it's very important that we are scientists and we should treat ourselves as equal always and focus on the scientific discussion in as our higher and unique priority. We have a map here, and probably I don't think that I need to comment very much, but we are here in this room. We have the exhibitors here, and also we have all the indications for that are present, and also some of the exhibitors in this area. Please visit them in both of the room. If you need to go to the toilets, they are downstairs and there are many indications. There is a John Lennon picture and Madonna as well. Between John Lennon and Madonna you find the toilet downstairs. And also for, we have for the Wi-Fi, probably you figure out already that Una Hotels works without a password. Food is permitted everywhere in the rooms, and please help us to keep it clean. Please be sure that you have all your property with you all the time. Do not leave computers or phones here when you go for lunch or so, because we try to ensure that this is a safe environment, but just keep you valuable with you, and also because we all need to charge our phones and our computers. On both sides there are many chargers, so on those red tables, so please do not hesitate to charge your device there and watch that they are safe. And okay, I would like to also thank the program committee, apart from Gary Bedr, Maz Haniffa, Shian Prabhakar, John Randell, Aviv Regev, Lucia Robson, Sarah Teichmann, and Alexandra-Chloe Villani. Apart from myself, that I've been working for writing, for organizing this meeting and the program. As I mentioned briefly before, so anyone can contribute to the Human Cell Atlas. It's not a closed consortium that started years ago, and it's not a club where it's difficult to enter. Actually, we welcome many collaborators, many new collaborators. Everyone can contribute, and you can join the biological networks and any other initiative by being here at the meeting. So please do interact. Do interact during the coffee breaks and all the other occasions that you will have, or the poster sessions and so on. Please see how because we need. This project is a very large and complex project. We need everybody, and and many different expertise will contribute to this fantastic atlas. If you are a member of the biological network, you will be helping to select a data set for the integration, to implement a human cell atlas bionetwork roadmap for your organ or tissue of interest, participate in cell type annotation jamborees for your network, analyze and integrate the atlas data, and join bionetwork meetings and collaborate with a researcher in your field. And all of this, of course, will help to integrate a lot of new data, new ideas in your research. And yes, those are the network for the various tissues. You can scan the QR code as well if you would like to have some more information, or go to the website. And I invite you to visit the website. So take a second if you want to scan this, or go to the humancellatlas.org. And in this meeting, we have several goals. This is not necessarily a meeting where we will hear only presentation, but we have targets that we would like to achieve as a consortium. So we would like to discuss the state of the human cell atlas, work for the biological networks, and hear the updates. Define spatial biology roadmap. So we have been working a lot on single isolated cells, and now we need to work also on spatial to really understand the relation and communication and many different relationships between cells. We want to work for a roadmap for advancing equity and diversity in human cell atlas. It's not only Europe and North America, but we are already working with all the continents. It's good to see many Asian colleagues and South American that I've seen already, and we will continue to involve and include many more, and also finally for the computational analysis. The schedule for today is that we're gonna discuss the state, and I think that I don't have to read the whole schedule, but yeah, please just leave there for a few more seconds so you can have a look at this. And also we're gonna also be here tomorrow from 9 a.m. and we particularly gonna have various parts, but particularly on the computation, computational biology in the afternoon, and discuss and wrapping up tomorrow by 5:30 p.m. There's one more point. Actually, we have on October 1st we have a follow-up meeting. It is not going to be here in this hotel, but it's going to be at the Human Technopole. That is after the last station of the Red Line. The other side, where is the Human Technopole campus, you're welcome to join that as well, where we're going to have the Human Cell Atlas Middle East meeting, and also Cell in Context Symposium, which is essentially focused on organoids as one of the focus of this meeting. So, and I think that we are ready for many very nice presentation and a discussion. Thank you for your attention, and going to probably Aviv and Sarah for their update. Thank you.
It's just fell. Sorry. Can you hear me? Yeah. Happy Sunday morning, everybody, and a very warm welcome to this year's Human Cell Atlas General Meeting. Aviv will be joining me as per tradition, and I'll kick off with an overview, as we always do. Hang on, how does this work? Next. Is this? Okay. Next. Woohoo! Yeah, we're away. As Piero said, we're an open community. I loved the introduction and the spirit, and really looking forward to the next days where we're discussing. We developed this mission statement about 8 years ago, And the idea is, of course, that we create this reference map of the cells and tissues in our body. And that's really what we are here today to kind of take stock and then plan for the next phase. And a reference map means that we want to have, you know, a high quality and definitive atlas, and that's what Aviv and I really agreed on from the day dot. Okay, so we want to have full body coverage. We want to have high quality integrated reference objects where we have collaboration between the biomedical experts, the data science, AI/ML, and so on. And that provides then a foundational resource for the biomedical community, you know, across academia, across pharma, across biotech. This is really like the Human Genome Project, a foundational resource. And as Piero already said, from the day dot, we've emphasized diversity in both the data and the scientists that participate. And that was very nicely kind of articulated by Piero. Hang on, I don't think I need this bit. Or is this? No, that's not working. That's not working. Okay. So what do we mean by a reference map? Well, where we stand at the moment is obviously, as you all know, is we've got tens of millions, even hundreds of millions of suspension cell and nuclear data points particularly for RNA. And so what that means is that we're now at the stage where we can develop a reference map and reference objects for the cells in all the different tissues in our body. And one of the things that we want to kind of focus on and lay out and discuss in this meeting is how to build an atlas, how to achieve those reference objects, basically both on the level of data generation as well as the data integration, which is now so important and you know, an ongoing project simply because we've had you know, over the last phase, sort of building up to phase 1 of the Human Cell Atlas, so many different data sets generated with different technologies in different labs around the world, collaborative projects. And so the data integration and you know, building the reference objects is actually a technical challenge. And so what we've been doing as a community is developing guidelines in terms of making atlases that are robust to technical factors, incorporating all the landmark studies that have come out and that are in the public domain at this point in time. Obviously, this is a data science task, but it has to be driven in a close collaboration with the biological networks who represent the biomedical scientists. So it's a kind of iterative effort between AI/ML data science as well as sort of the scholarship and the knowledge in the community. And that leads to annotations with those expert inputs, and which we'll iterate sort of within the cell annotation platform. And we'll have more presentations on that later from Chloe, who's sitting here at the front. And the end goal is, of course, to have those integrated data objects with metadata as well as the code publicly available. And so that's thanks to the collaborations of the analysis working group, the data science community with the biological networks. And just to show the views from these biological networks, so we have 18 at this point in time that have kind of consolidated basically and come together in the community. You can see all the different 18 networks here, and the numbers of cells that are kind of in the work, so to speak, for building reference data objects for all of these biological networks. In total, there are over 100 million cells or nuclear data points, and the spatial data is sort of coming along very nicely at pace for all of these. Just in case for awareness, there are 4 completed version 1 assembly objects. The lung was the first, and that's actually going on to version 2. The nervous system, so the brain Bionetwork, as well as the eye and first organoid atlases. And these are the 2 new ones that are going to come up at the data portal on data.humancellatlas.org. map.org. And we're on track this year still to have immune kidney and liver objects as well as gut. So that's, really exciting. I think that will be, an incredible output of our community as a foundational reference resource going forwards. And I really do feel that this is very important. We need to take it very seriously, because this is where the whole biomedical community will come to know what the cells are and what their molecular fingerprint is. So it's really an important exercise, an important output that we can offer also from our project. We're now at over 420 source data sets, 60 million cell profiles, FASTQs and matrices, of course, with the key, and I want to give a big shout out to Chloe, Chen, and others for harmonized metadata and tailored biometadata as well as diversity metadata. And these metadata, it's just a word, but it's actually so key that we, from an interpretation point of view, that we know where our data comes from. And that's actually, something that we've tackled very early on in this project. And that will add a lot of value, basically. And the Human Genome Project was different in that sense, right? We're kind of thinking ahead, I think, very quickly, and putting out the data with the metadata about the, as much as possible about the donor origin of tissue. So just to emphasize the resources here in terms of both ingestion and serving, is that we've evolved this at this point in time from the original. Those of you who were here early on remember the data coordination platform. We've now got a matrix store that uses cell by gene, a data repository, and we'll hear more about the cell annotation platform. So these three components help us to make an assembled atlas, and then serve the data on the what we're now calling data portal. So the metadata standardization here is through different tiers with different levels of detail, basically, in terms of metadata, and the minimum is sort of the technical information, so that we can have a better grip on batch effects and so on. The second is with more biological information for downstream analyses, and then there's obviously the annotation kind of level, which CAP kind of can support us in developing the reference for all cell identities and have consensus annotations also, because this naming is obviously an important, is an important sort of component of our project, so that people know what they're talking about and using the same words in a controlled vocabulary with ontologies, et cetera, et cetera. And that's kind of an ongoing thing, and we'll learn more later. This is just a short animation, but there'll be much more detailed information about cell annotation coming. The data portal is really the home base for the cell atlases, and this is where you can access all the source data sets and these reference objects as they're being released in their version 1 form and as they're coming forwards in version 2. Very exciting news flash, is that we now have HCA's Managed Access Service and a Data Access Committee. And Aviv has pushed for this actually from very early on. This is we're finally there, and massive shout out to all the teams, who've worked on this from the early DCP grant and the support that's made this possible. And hand in hand with that, we now have launched the HCA Stichting, which is key because of general data protection regulations in the EU and facilitates sharing of EU. So we now have 2 not-for-profits, HCA Inc., which Aviv and I co-chair the board, and John Randell, who's the Chief Alliance Officer of HCA, is the director of that entity, and HCA Stichting, which there's a separate board for, which is now starting off. And it's really thanks to all the directors and the staff members also of the executive office, who are sort of the backbone of this, and in the Netherlands, Martijn, Musa, and others who've supported setting this up, which is a big step forwards for us. I will now, as per tradition, hand over to Aviv. This is the laser pointer, and then you go forwards.
That is the famous
with
laser pointer.
Yeah, there you
Keep
go.
my distance from you. I don't have COVID. I tested. I follow the rules. Unfortunately, I managed to get myself my first common cold since 2020. Because once the pandemic started, you know, people started putting on these masks. I actually haven't experienced the common cold. So I think it's really exciting to see that first version of the map. And I think it makes you begin to think like, you know, 100 million cells profiled. They're being assembled in these 18 atlases. They're being provided on a data platform. You might have noticed the dates 2025, 2026, which if you can add the number 10 to 2016 actually makes sense. So we will have ourselves a reference map of single cells. And this could lead you to think of, fine, our work will be done in a couple of years. Well, kind of not so fast. And so I want to draw your attention to the rest of our mission statement. We handled 2 words, reference map, but the mission statement has a whole bunch of others, and we're going to spend the next 20 minutes or so parsing them out. So the next one is the word comprehensive, and that's actually going to be the crux of the matter. And that goes well beyond individual cells, RNA profiles, as much as we might all love them so much. Because, for example, there's many other profiles that one would measure in individual cells at the multi-omic level. Of course, we need to position them spatially and at multiple levels of resolution, and we have to pay particular attention to human diversity, otherwise we wouldn't be able to call it the human cell atlas. It might be a human cell atlas, but not the human cell atlas. Now, we're actually working at all of these levels. The ultimate atlas will cover all of these. But at this moment in time, we have a particular focus at the spatial level. And this is because, first of all, their time has come. The technologies have matured to the point that we can really do this work. And also because we need to do this at multiple levels of resolution, from the subcellular to the histological to the anatomical. Otherwise, we won't reach the scale of the human body. And in order to build this comprehensive spatial layer of the atlas, there's 3 key threads that are ongoing in our community, and I want to draw your attention and your energy to them. The first one focuses on the lab technologies themselves, assessing them, working with them. We have a little bit of an embarrassment of riches, as is our tradition. We don't just use one method, we use them all, but we need to understand them, their relative strengths, and how we deploy them for our problem. Those of you who have been in HCA meetings going back to 2016 know that it was the same with the single-cell profiles. I still remember the vigorous debates about the nuclei. Yeah, they were vigorous. There were debates. We now realize sometimes we do cells, sometimes we do nuclei. And it actually all works out. A second focus is on the algorithms and the pipelines that we need, and we need them in order to guide the sampling, the data collection, the analysis. We need to develop them, and then we need to apply and deploy them, and then we learn lessons from that, and then we develop them more. And so all of that effort takes a substantial amount of effort. And then, of course, the third and most crucial thread is that we need to collect more data. You saw that there were already 17 million cells collected by spatial methods that people have reported into the census. There's actually a lot more of those in kind of the public realm, and this is because in a single section, analyzed by spatial methods, you collect a lot more cells than we have historically been used to, but that is just from a single sample. And so we need to make efforts to define the right marker panels for signature-based methods. We need to obtain the right samples. We need to perform the experiments. We need to integrate with other views like histology, anatomy, and map to a coordinate framework. All of these threads are supported by members of the HCA, members of the Standards and Technology Working Group, or STWG, members of the Analysis Working Group, or AWG, the biological networks, you know, each and every one of them. And this growth of the spatial atlas has actually become rather rapid now. It's going to be really apparent at the upcoming HCA publication bundle, where there's already spatial atlasing along with cells and nuclei for the GI tract, the thymus, the placenta, embryonic skin, skeleton, and limb, and the endometrium. And we're going to see more and more of that. Now, concomitantly with data come the analysis tools. They go hand in hand. And we're seeing rapid progress of computational tools and methods for spatial purposes. So, for example, the Web Atlas pipeline, these are just illustrative but cool examples. It helps integrating spatial and single-cell profiles. There's multiple methods for that, but they're becoming more and more mature and in routine use. And then there is the ELD. I really like this because it's called the effortless detection of landmarks, which allows you to find the landmarks and do histology registration. These are two of a large frame of tools, but there is a little bit of a what if that I find to be particularly exciting. What if we could move from computational efforts that are really post hoc analysis and integration, meaning assembling all of the data that we have, into a generative model? That means a model that we learn from data, but then can generate new data for us, or can become a common coordinate framework for other data, even from other modalities that were not used in the model initially. Why is this so exciting? Because it might liberate us just from collecting data in perpetuity. So, for example, if a model like this is successful, we might be able, once we have an atlas model in hand, to take a new sample from a new individual where we have only histology, and then use our map, use our reference atlas to generate for us single-cell profiles or spatial stains virtually without doing an experiment. Now, that's not science fiction. So here are just two of multiple examples that are already making their way out there, called CAST and SHAF. So CAST, for example, from Xiao Wang's lab, uses single-cell spatial, histology, and anatomy data. They did it in a mouse brain, where you have the most these days, in order to learn a reference map. And this reference map is not just an aggregation of the data that they have. Once this map is learned, you can take any sample with these or other modalities and map it to the reference, a new sample. And once it's mapped, you essentially know where it came from. So imagine you had a little brain biopsy, and all you did was profile nuclei from these biopsy. You did no other measurement. With this map, after the mapping, you will know what it looks like histologically. You will know where it is anatomically, even if you don't know any brain anatomy, and you will know the spatial location of all of those cells. SHAF, which was developed by Charles Comiter and colleagues, is a generative model. It was trained on matching histology and single-cell data, like single-cell RNA-seq. And once you train it, it can receive just the histology data. Before I gave you nuclei, now I'm giving you histology. Only histology data, and will generate for you single-cell data and map it to the histology. So in this way, the generated atlas is not just a description of data we already collected and measured, but it is truly a comprehensive model of the data that could exist. Now, once we have a map, we use it to understand biology, and understanding biology requires effort. This is where querying the atlas becomes absolutely essential. In order to query the atlas for understanding, we usually come into it with something we've done. For example, a separate data set. It can be from an in vitro lab experiment in cells, it can be from an organoid, it can be from an animal model, it can be profiling of a patient cohort which has a particular condition. None of this is the atlas per se, but that's the query. And so it requires taking these query data and asking questions of the reference map. It's kind of analogous to how you come with a new sequence and you want to search it against the human genome. But, we just do it with cells. So with reference mapping, you can annotate cells. You can ask how the composition and the states of the cells in my sample relate to the reference. And you can ask how it might be organized in space and more. And again, we see a growing number of tools from the HCA labs for this. So for example, CellHint automatically harmonizes and integrates cell types. Covet and Niche Compass characterize and identify cell niches in spatial data. There's Multimil. Multimil helps query multimodal single-cell data using multiple instance learning. Just illustrative example, but it's reality. Now, as these data grow bigger and bigger, income foundation models. Foundation models help us move from individual atlases, like Sarah showed you. We have an atlas for the lung, and we have a separate atlas for the gut. These are invaluable, but we also want a unified model of the entire human body. And in fact, if you look across biology, the Human Cell Atlas is one of the only data resources in biology right now, along with DNA sequences, protein structures, and histology stains, which are really fueling the work of foundation models for biology. I think the members of this community can kind of pat themselves on the back for being very prescient a decade ago, when no one talked about models like these, realizing that we should collect data that allows us to learn these kinds of models. And these models are growing very rapidly. This is actually a slide that we showed last year. This was the first generation of models. They're already becoming, rapidly obsolete, as, nothing in this field lasts for more than 6 months. And so they worked at the scales of tens of millions of cells. They were already tackling kind of classical tasks like annotation and gene program discovery, and they were starting to dabble in these new tasks like predicting the outcome of perturbations or searching for entire cell profiles. In the past year, we're already seeing a series of exciting additional developments. So there's models that scale now not to tens of millions, but to hundreds of millions and billions of cells. They operate across species, like these universal cell embeddings, and multimodal LLM-like models, including niche former that does single cells and spatial data as a foundation model. There's also vigorous work on benchmarks, like SC-eval, that help us understand what complexity of model do we need to have in order to achieve models that are truly performant on different kinds of tasks, like annotation or predicting the outcome of perturbations or predicting cell types that we didn't measure. The scale of the data and the initial successes have really started inspiring a new community of people that are starting to say, well, we can imagine virtual cell models, even of the inner mechanistic works of cells and tissues that pushes beyond the vision of the Human Cell Atlas itself. So this is really an exciting direction. We're seeing it growing. There's a lot of open questions that remain, but I believe we will see a lot more coming from this. And while this is happening, and the foundation models are solidifying, we're also beginning to see the next generation of approaches already emerge to help us make sense and navigate these massive atlases. And they include cool things like autonomous agents that cannot just engage like a chatbot, engages, where you ask them a question in human language and they give you some answer, but these agents can plan, they can take action, and they can access and deploy analysis tools for you. So for example, and this is real data, an agent can get an unannotated single-cell RNA-seq from a data set from a user, and the user can ask a general question like, what are the cell types in this data set? And in order to provide the answer, the agent would figure out autonomously the analysis tools that they need to deploy in order to annotate cells. They will spin them up, they will execute tasks, they will collect the information, they might iterate a few times, and they'll come back to the user with an answer of what are the cells annotated there. So watch this space, because a lot more will be coming. Now, all of these bring us to the last component of our mission, which is to use the map for diagnosing, monitoring, and treating in health and disease. And we increasingly see our reference atlas being used by a very broad community of people, some here and some not here, in order to play a crucial role in disease studies. And that includes things like basic disease mechanisms, mapping GWAS genes to function. Christoph asked me a question about that, Christoph Bock, and even direct therapeutic discovery and making regenerative medicines. So for example, there's recent analysis in Alzheimer's disease from Phil Dieger, who's a neurologist. He relied on large single-nucleus RNA-seq atlases to tie in specific cells and gene programs to a causal chain from beta amyloid to tau tangles to cognitive decline and dementia in Alzheimer's. There's foundation models that were proposed to predict genetic manipulations that would be needed to yield cell therapies with desired specific states, for example, from Bo Wang. And in IBD, there are now clinical trials with targets that were chosen in a manner that was informed by single-cell atlases of the gut. And this kind of success, there will be many, and they will not be ascribed to us. They will be ascribed to all the people who use a reference, just like the human genome is used by plenty of people every day. And the people who have sequenced the human genome no longer keep track of its usage, because kind of it's just being used. This kind of success, for us, opens a unique opportunity and a unique responsibility to make sure that the healthy atlas is done the right way so that it is specifically used for diseases with a high global disease burden. And those are diseases that would have, unique risk profiles in specific geographies and specific populations, but that generally tend to be underserved. By that, I don't count IBD, not cancer, and not Alzheimer's disease. This could be conditions that afflict many, many women around the world in childbearing age, like endometriosis. These could be oral cancers that are caused by tobacco chewing and are very prevalent in India. Or it could be tuberculosis and other infectious diseases that are major killers worldwide but don't get nearly as much attention in terms of research. This effort is really essential for HCA to be true to its equity values and its commitment to humanity, and also for HCA scientists, even for the Healthy Atlas, to be able to successfully engage on a global scale. Okay, so let me summarize what's next. We're on track to complete the first version of the atlas based on single-cell RNA profiles. You saw everything under the hood from Sarah. We're improving the use of the atlas through algorithms, platforms, portals, and building it as a foundation models of cells. As we move into the future, we're now fully building the comprehensive reference, especially as a spatial 3D maps. And we have to ensure that our atlas is globally representative, especially by including it as a reference for diseases that have a heavy global burden. And this is not done by one lab, it's not done by one institute, it's not done by one country. This is done by basically all of us. Not just the people in this room, the people in the room, the people online, the people who didn't have time to sign in for a meeting but are actually doing the work. And it's across 101 countries, almost 1,900 institutes, and more than 3,500 members all over the world. Those of you who used to be in HCA meeting years ago might have remembered that the bottom part of these pictures was embarrassing in the beginning. There would be single digits there. We're no longer there, and that's thanks to the really unbelievable work from the regional networks in Asia, in Latin America, in the Middle East. There's a meeting, October 1st, for the Middle East Network, and in Africa. And we really see the difference. Now, our members have done so much. They've done so much that they've published 445 papers to date. And you should stay tuned, because at least 33 of them are coming up in our publication bundle that is going to span atlas integration, development, and methods papers. You should watch for them somewhere in November across many Nature journals. You can talk to Ellen Todres about this. Ellen, wave. But also, Michael Fletcher from Nature Genetics is supposed to be somewhere here. He's way back there, people. So if you want to ask the Nature editor, he's your guy to ask these questions. Our community is made of many scientists, including the many, many scientists among our funders. And that includes the Chan Zuckerberg Initiative, the NIH, the Klarman Family Foundation, the Wellcome Trust, the ERC, the Helmsley Charitable Trust. Thank you, Helmsley, also for helping us with this meeting. And many, many, many more. Our partners in many consortia, HubMap, BRAIN, Phantom, many disease consortia, KPMP, HTAN, and many, many, many others, from cancer to IBD, kidney disease, and more and more. And I'm really pleased to say that more recently we added 2 industry partnership programs. There's one for pharma and biotech, and that is in order to ensure that we fulfill HCA's goal to impact translation to medicines, and this will help support our meetings and capacity building workshops in the community. And we have a new commercial discount program, hurrah, hurrah, thank all those great exhibitors with many, many key vendors, including some new ones and some long-standing partnerships, to help members stretch their resources to the max so that we have the most data and most analysis capacity for our atlas. All of this is done by the people in the meeting. It includes our organizing committee members, the leads and members of the expert working groups for analysis, ethics, equity, technology, and data ecosystem. And in each of the biological networks, each of these 18, it goes from dozens to hundreds of members in a single biological network. They're organized by the organ and the system. And of course, we're all really grateful to our amazing executive office team. They really make this massive global consortium run every single day. Piero and I were kind of chatting before, and he said, it's actually pretty easy to organize these meetings. Because, of course, it's not easy at all. It's just that they do all the work. And so I actually think we should take a moment and ask them to stand up and thank them. So Aya, Yoshi, Esther, Anya, Anne, Bridget, John, Lucia, Jenny, Christine, Ellen, and Sam. And I asked you to clap them, not just run after them when something goes wrong. But of course, they're going to make the meeting run like unbelievably well. Thank you all so much for everything you do. And so, all of us, I think, come here because we truly wish to make a difference. We want a global initiative that benefits humanity. We want it to benefit all humanity. We want it to be anywhere and everywhere. And that kind of happens one workshop, meeting, training, experiment, analysis, algorithm, annotation, project, conference, session at a time, all across the globe. So let's have a fantastic general meeting, everyone, and enjoy our next 3 days, and then go back to assembling our atlas. Thank you very much. Leave for a few questions, if people have them. I'm looking at John. It says 6 minutes and 42 seconds. So I'm like, that's a first. Any questions? And if not, we'll just continue. There will be plenty of panels and activities going forward. Apparently, there is someone. Oh, yeah, I see. I should keep my distance from her. I don't want to give her a call.
Thank you for this very great overview. I think it's super cool to see how the atlas will be built from transcriptomics and expands to spatial and so on. I was wondering, do you also consider taking functional readouts into the atlas? How is this in the scope?
I mean, I'm happy to take it kind of, so I would give two answers to that. So one is basically, in my opinion, a map gives tremendous information about function, okay? So if you have a map of tissue architecture at single cell resolution and full molecular breadth, basically that gives deep insights into function that we never had before in terms of cell-cell communication, right? So the molecular receptor-ligand communication that's signaling across cells in homeostasis, the control of cell identity and cell state. In our healthy body, there's constant regulation happening from progenitors to differentiated cell types that we learn by mapping them, the molecular process. And of course, we have the Human Development Cell Atlas that's doing the same thing through embryonic and fetal development. So if you think about protein crystallography or structural biology, cryo-EM, then basically what we're doing there is solving the atomic coordinates of each amino acid in 3D. And that technology has transformed biology by learning about the atomic and molecular details of protein function. What we're doing here in the Human Cell Atlas is solving the structure, the molecular detail of the human body. And that gives deep insights into function. The second thing I would point to would be the disease atlases that Aviv talked about. So we always have, like for all the tissues, for immunity also, we're baking the disease into the annotation to some extent. But for every tissue, obviously, there's the disease counterparts, which can be for many different diseases. And there, I mean, the function is inherent in the sense that you're seeing the delta, the difference in disease. I don't know, Aviv, would you like to add anything on top of that?
So I think Sarah covered really well the fact that you can infer a great deal of function, or putative function, as I would call it, from observations. That is absolutely true, and this is because of the structure-function relation in biology. When things are structured, and I don't just mean in physical 3 dimensions, but in the relationships between variables in biology, they tend to actually have a functional meaning, especially if the structure is repeatable. There is a second layer for function, which is what we think of as causality, right? The relationship between a perturbation and its outcome. That requires genetics, and that can happen in 2 different ways. The first one is through human genetics. The atlas, because of its emphasis on diversity, is gradually moving into the human genetics realm. And in the human genetics realm, you get function through causality. The second one is the perturbations that we do in an engineered way. And those do not happen, I would say, through the human cell atlas per se, but it is one of the emerging efforts in the broader community that intersects very highly with the people who are in this room. And these kinds of perturbation atlases are increasingly emerging, and they become a great counterpart because the readout, the phenotype you look at, is actually the same kind of phenotype. So now you can get to those causes, but of course that doesn't happen inside the human body. It happens in a dish, in a cell, in an organoid, in an animal model, but it doesn't happen inside the human body. That you only have with human genetics. And the third one, and even within that layer, you have a virtual version of it. I referred to it when I talked about the foundation models. We increasingly find that they might be performant enough to actually correctly predict the outcome of these genetic interventions. And if that would be the case, then in fact the atlas would have even more value to it than people might have anticipated initially.
I mean, what's also worth mentioning is the meeting on Tuesday.
Meeting on Tuesday. Come on Tuesday.
They organized Christoph.
Cells in context.
Yeah.
Christoph, where are you? There you go.
So the organoid systems that are the counterpart of the Healthy Reference Atlas will be the topic of the organoid Bionetwork, and there's a dedicated meeting on Tuesday. And so there's a kind of in vivo, in vitro component of the human cell atlas that we have baked in.
And then the third, and very briefly, is you have the mechanistic layer, because the causality I described is only genetics. The biologists like mechanisms, they want to know how, not just whether. And the multi-omics view is actually essential for that. So I would say there's a lot there. It doesn't solve the whole problem. You need to also have bounds. We also can't characterize all diseases. But there is a very nice relationship and overlap, and it's a big enabler for understanding function. I think we'll leave it there. That was kind of a meaty question. Maybe there's time for one more, and then we'll stop.
Hello. I want to firstly thanks that you give us the opportunity to join this great meeting. But I was wondering how this atlas will cover all of the populations, and how can it serve all, populations in the world, because in terms of anthropology, there are lots of minor populations across the world, and maybe there are just a few, hundreds of people living in some specific areas of the world. So you will cover all of these.
You don't cover all the populations. Sorry. First of all, you do not cover all individuals. You need to cover representative samples up to a certain level of, I would say, ancestral variation. And that has always been the story in biology. Biology is ever diversifying. Every second an individual is born, they have a unique genome. And the point is not to liquefy them and analyze all their cells, obviously. So that is not the purpose. The purpose, as is usually the case with variation, is to capture enough of it to know that you can generalize from there, and also to understand up to which point does it get. So I'll use by analogy the efforts of our great human geneticist colleagues, and there are actually some great human geneticists in this room today. They have not characterized all human variation. They have characterized it up to a certain level of rarity, and they're also quite aware of what they don't know. They know that they have some populations that are better represented than others. Our goal is to try and do a little better in terms of representation up front, or maybe a lot better, but we know that it would be a fool's errand to try to say we have covered everything. We're trying to be reasonably representative. And I would encourage you to talk to Shyam, to Mark, and to others here about this. Sham seems like he's chomping at the bit to say something, so maybe we'll give him the last word, and Sarah and I will graciously go off stage at this point.
Okay, just quickly, great question. I hope you can come to the Diversity Task Force and the GDN session tomorrow, and the breakout sessions for that to discuss this more in detail.
Thank you very much, everyone.
That's fantastic. Thanks. Thank you, everyone. Yeah.
Good morning, everyone. My name is John Randell. I'm from the HCA Executive Office, which you just met. One thing I want to really stress about the Executive Office is that those individuals that you saw are actually spread across the globe. We have an office in Singapore, in Japan, in the United States, and in the UK. And I'd encourage you to reach out to any of us, either at this meeting or over email. You can actually click on our bios on the webpage and get our email addresses if you have any questions about HCA or if you need anything. It is my pleasure to introduce the next session, but first I want to actually add to Piero's thanks to all of you for being here and to our sponsors and exhibitors for making this meeting possible. I also want to thank Piero, who is continuing a strong tradition of excellent meeting hosts in the Human Cell Atlas world. So thank you very much to you and your colleagues for everything you've done to make this meeting such a wonderful experience already, and certainly over the next 2 or 3 days. For the rest of this morning, we're actually going to have several sessions that will dig into more detail about many of the themes you've already heard about from Piero and from Sarah and Aviv. They include, of course, the scientific scope and the rigor of the atlases that we are developing. It includes the communal nature of our work, the fact that we really depend on all of you to achieve that sort of scientific quality that we're really looking for in the products that we are creating. And finally, really, I want to emphasize the adaptability of the HCA approach. And there are a couple of different dimensions of that. One is certainly the adaptability over time as we continually learn and improve upon our processes. And Chloe will have a lot more to say about that now. But the other is the adaptability between tissues and systems. So we're really adapting our approach to the specific needs of each biological network and each of the research networks that's contributing to the atlas. And we're also working with a lot of other consortia in these processes. And you'll hear about that at the end of the morning in a session focusing on the work of the biological networks, and in particular how we're organizing the networks for atlas assembly. This next session is really a two-part session. So we'll first hear a more in-depth summary of how we are going about building atlases, where we are in that stage, and what we're learning along the way from Chloe Villani. We'll then have a coffee break. We'll come back and we'll learn more about how cell type annotation is being performed in the HCA context using the HCA cell annotation platform, specifically looking at the gut and brain atlases as an example. So every time I introduce Chloe Villani, I have to add titles to my introduction, and it gets longer and longer. So I'll try to keep this very brief. But Chloe Villani is an institute member at the Broad Institute. She is a principal investigator at Mass General Hospital in Boston and a faculty member at Harvard Medical School. And in addition to being a really talented immunologist, she also wears many hats for HCA. She is a member of the organizing committee and the executive committee. The organizing committee is a group of 36 scientists around the world that really provide the scientific direction for HCA. She is a co-coordinator of the HCA Immune Biological Network. She's a co-chair with Shyam Prabhakar of the Data Ecosystem Oversight Group. And finally, she is the lead PI on the cell annotation platform that you'll hear about more after the coffee break. So I'm very pleased to introduce Chloe, and thank you again for attending.
Thank you. Yeah. Buongiorno a tutti. It's such a pleasure to be here. Yeah, that's the kind of the Italian for this morning. It's such a pleasure to be here today. The HCA General Meeting is my favorite meeting yearly. I get to see my friends from all over the world, make new friends from all over the world. I look forward to meeting a lot of you. So we get to spend a bit of time together this morning. And I hope by the end of this morning's session, you have a better sense of how we've retooled some of the data ecosystem. What are the toolbox available to you? Who are the people that are there to support Atlas Assembly that you can reach out to? I want to first situate ourselves in this timeline, right? Aviv mentioned it was funded in 2016. We spent the last few years building our grassroots initiative, right? Developing technology, benchmarking technology, generating our first atlases, expanding our community across different borders. A very exciting time. We've now moved into this new phase of atlas assembly. We're right in the middle of it. It requires a new way of approaching it, because it is one way for each and one of us to build our own individual data sets for our own academic endeavor and publish them. It's a whole different story to bring them all together, to produce a product that can empower advancing biomedical research. This is where we are right now. Last year we had the first lung and brain atlas published. We just got the retina and the organoid atlas approved. It's an exciting time. Every one of these 18 biological networks are now working towards their first version, and in some cases second version of their atlases. And so there's a lot of atlasing efforts out there. I'm going to spend a moment explaining how is HC different from some of these other ongoing efforts. So we are working towards creating a gold standard reference atlas. What does it mean? We are taking a consensus community approach, and that is not trivial to do. We're applying rigorous standards. We're going to go over some of them today. We're operating at an unprecedented scale. Just for version 1, we're well over 400 data sets, and we're aiming to really have a global representation. Diversity is at a key core of our HCA principles. And so, when we talk about version 1, this is not the end of the journey. It is very much the beginning of the journey, right? And so we're hard at work right now. And we must continue iterating in the next decade so we can build the most comprehensive atlas out there that will push forward some of our biomedical endeavors. It is challenging to build the first version. We cannot take any shortcuts because this becomes the foundation on which we're going to continue building our atlases moving forward. I remember last year sitting on this stage where Chen and I had a fireside chat. Of course, we had different commute challenges, so he was on a big screen. I kept on looking at you, Shyam, and we were talking about lesson learned from version 1 of the atlas. Right? We organized across 3 waves. We're part of wave 1, and there's like the concept of building an atlas, and there's actual doing at the assemble. And we realized there was a lot of unforeseen challenges that we had to overcome. What I'm really proud to say is that over the last year, we went from talking about these challenges to finding solution, right? And so if you haven't heard a lot from us, it's because we're hard at work in trying to define new toolbox, new procedure, centralizing our effort, and implementing strong governance so we can continue iterating these atlas assembly. And that's partially what we're going to talk about this morning, so that I hope at the end of this talk you have a better understanding of what we've done. Aviv presented this. There's a new family member on there, and so Sham and I now co-chair the Data Ecosystem Oversight Group, which we're going to talk a bit about this morning. It's a new addition to the group. And so there are a lot of key advances. So I'm going to spend the rest of the talk going through details of these 4 new features. So we created this Data Ecosystem Oversight Group. We have now an ecosystem that is enhanced and tool to develop atlas at scale. We're going to go over some more details about the GDPR compliance and end on discussing how we want to have more engagement from the global community. So what is the scope of the DEO, or Data Ecosystem Oversight Group? We provide oversight and coordination of the HCA data ecosystems activity as a whole. That includes the data ingestion, the data storage, the integration of the data sets, self-type annotation, and the HCA data portal. We receive regular updates from different project leads across the different pillars, and we provide recommendation to the organizing committee, and we liaise with other working groups. That's a better way of summarizing what I just described in terms of governments, and we help in supporting developing policies that will feed in the infrastructure, which we're going to go over in more detail. That includes the metric store, the cell annotation platform, the data repository, and the data portal, and this is done with related governments from the other working group. We're extremely grateful from members of the community that have agreed to join the Data Ecosystem Oversight Group that I co-chair with Chen Pragbakar, and we have members from around the globe that are participating in this committee, and also across domain expertise, biological biological network representative, experts in different fields of the analysis working group and IT, among others. And of course, members of the executive committee, so we can liaise more efficiently. I want to now spend a bit of time on the data ecosystem retooling that we've done. I'm going to go over metadata standardization, some of our ingestion redesign that you saw quickly in the introductory remarks, the HCA data portal. In the next talk, after the coffee break, we'll go in greater detail about the cell annotation platform. So it is, as I mentioned earlier, it is one thing for each of us to work in our own lab, generate single cell data, publish a paper, and move on to the next stage. And it's a completely different story to try to start bring all of our data together. It requires us to start standardizing the metadata. If you don't standardize the metadata, if you don't standardize your file format, it becomes impossible to bring them together. And that's what we're trying to do with the Human Cell Atlas, right? And so it's important for the data integration to standardize these fields, and it's important so that we can build software to be able to query and navigate the atlas moving forward, right? There's no way we can build a portal if we have 15 different file formats when assembling these atlases. And so what we've done is that we try to simplify to some extent, yet keep it comprehensive enough so that we could achieve all of these goals. You saw them in the first talk. We regrouped them through 3 different metadata groups, and we're going to go through them in slightly more details. The first one, we call it Tier 1. It is common to all 18 biological network. It is meant to be completely open access. It focuses on technical field and broad demographics that are tied to your experimental design, as well as broad analytical parameters that you might have used. So for example, simple ID and processing, broad demographics, no identifiers that would be like, age range, for example, experimental detail, sequencing information. And this is led by the Cell by Gene team. Cell by Gene, of course, has evolved their Cell by Gene schema. We've worked really hard with them over the last year to develop an HCA plus schema. There are about 39 fields that are well detailed. And they're in charge of your gene expression metrics as well as this Tier 1 metadata. Yeah, but you've heard about the Tier 2 metadata. These are fields that are going to be more specific to every biological network, except for the genetic diversity fields that have been developed by the Genetic Diversity Task Force, which are meant to be shared across all biological network. These can be managed access, and again, specific to each biological network. So for example, ethnicity, and I'm giving the example of Lung, who are currently working towards our version 2 and focusing a lot on COPD data sets. This is managed by the HCA data repository team at EBI. They can also handle the raw data, the FASTQ files. And finally, you've heard about the cell annotation platform. This is meant to collect the rationale behind your cell annotation, which we're going to go in more detail in the next talk, so that we can start comparing our annotation in a more objective way. And that includes some marker genes, some cell ontology teams, some synonyms, and this is led by the cell annotation platform team. And so, you don't need to worry about these 3 different pillars. We want to be transparent and tell you what's going on behind the scene. This is on the back end. Of course, on the front end, you have a more uniform team working together with these 3 different pillars. So I want to show you some of the face, because many of them are in this room. And so the HCA Matrix Store and Cell by Gene, we're extremely grateful to Jason and Brown, who have been working really hard with the team to kind of put this in place, from the data repository group, Tony, Gabby, Ida, and Arsenios. From the cell annotation platform, Evan, Mary, David, and Jasmine have all been working really hard in making this happen. All of them over the last year have helped conceive this very detailed documentation. There's a QR code. You're going to see this QR code come up a few times in my presentations, where you get the detailed definition of every field, how to assemble your files in a more standardized way, and how to connect with our team. This is meant to be coordinated by your biological network coordinators and your integration team that will also be there to help you navigate, as well as the executive office. The different biological networks are hard at work in assembling these different tiers, both for Tier 1 and Tier 2, and some have already completed both of them. And so the goal is, over the next 2 years, for all of us to cross the finishing line. Once you have the metadata across all 3 buckets, you're ready for Atlas assembly. So a lot of people have been hard at work in gathering this information and integrating the data. And so I want to do a quick shout out to the core integration team and of course the broader biological network champions that have been working together to create these first versions of Atlas. Once you have an Atlas assembled, it becomes available on the HCA data portal. You saw a snapshot in the first presentation. I want to give a shout out to Dave Rogers, who's been really working hard with us in tailoring the HCA portal, which to our needs. So the HCA portal is the front end where you can access all of this information. And so this is what it looks like from the portal. What can you access? You can access the end data objects for all these gene expression matrix. For the individual data sets that were used to create this integrated object, you can access to the full metadata tier 1, which is going to be open to public, and tier 2 through managed access requests. We're going to go through this. You'll have the hyperlink to get to the cell annotation platform to see the cell annotation, hyperlink to get to the FASTQ files, the matrices, and you can access managed access. So this is a screenshot of the long network. Every page will look the same. First, you get a description of the Bionetwork. On the far right, you get the different coordinators involved, and here at the bottom, you get an example of, a first integrated atlas. It tells you have an integrated atlas, how many tissue, normal and disease, the number of assays. If you click on the atlas, then you get more details. You get to know the individual data sets that were included to build this atlas, whether it was health or disease. You get to know, the number of cells that were included, and so forth. So this is the front end. So how do they all work, right? It may seem like everybody's a pillar. They actually work all together under the umbrella of the operations committee that has members from every single pillar, right? The matrix store, the data repository, the cell annotation platform, but also the executive office, the integration team, and the HCA portal. They meet monthly to make sure that they're all aligned. They use an atlas tracker, so I'm giving you a peek behind the curtains, through which we have the list of all data sets being included in the atlas. This is predefined by every biological network. And so this tracker is meant to give transparency to all stakeholders involved in the ingestions. It gives us a key point of contact for each atlases. It breaks down outstanding activities into specific tasks, so we can see when we meet the milestones. It displays the deadlines for ingestion, so we can see if we're on track or not. And it will link the source data sets to the different integrated objects, so that, for example, as the matrix store works with you on adding these tier 1 metadata, you can also work at the same time with CAP to add your annotation metadata. Some updates can be manual and some can be automated. And this is just an example of a tracker for the heart where you have the individual data sets. Some of them are published. I'm going to talk about unpublished data set in a minute. And you can see across different pillars where we are at. Is it in cell by gene? Is it in CAP? Is it in the data repository? And so it's important to know that for an atlas to be certified in HCA version 1 atlas, every data sets that contribute to this integrated object must have been deposited in every pillars of the ecosystem. So I'm gonna, you saw these criteria in the forms of graphics in the first presentation. I want to repeat them again because they're pretty key as we work towards these goals. So first, members of biological network working with the Bionetwork coordinators must have defined what are the landmark studies you think are critical for this first gold standard atlas you are building. And so they must include these landmark studies. The reference must be robust to technical factors that can affect atlas quality. That means we have to have tested for these tier 1 technical metadata. We need to be able to release them publicly so that for sake of reproducibility, but also for sake of expanding this atlas moving forward. Annotation must have been generated with input with representative of previous landmark studies. And so not only does your annotation must be in CAP, you must have involved some domain expert not involved in atlas assembly to kind of validate some of the earlier annotation. And finally, the raw data must be submitted in the repository, or at least we need to know where your FASTQ files are. We've been building a lot of documentations to help all of you build these atlases. Again, the same QR code will get you to the landing page where you get detailed information about our updated policies and new documents to be released very soon. I'm going to mention briefly unpublished data sets and briefly the standard operating procedures for atlas assembly. And so we quickly came to realize that a lot of you generously are willing to share unpublished data sets. Oh, this is getting exciting. This is how you make like a... yeah. Okay, well, we're lucky it's coming back. I mean, this is how you make sure you're all awake or getting you to sleep, I don't know. And so a lot of you are contributing unpublished data sets. I'm one of these examples, for example. And the question was like, so how can we make sure that you can still push your own lab's academic endeavor while still contributing to the Atlas Assembly? So we created detailed policies for unpublished data sets. You can read them in detail. And so we, HCA, want to protect unpublished data under embargo until the Atlas is incorporated and published in a scientific journal, that is peer review. And so if you're considering contributing unpublished data and you're unsure how to navigate it, please reach out to the executive office. Please read our policy about this. The access to the embargo data, the gist of it is that the access to your embargo data would be restricted only to the data wrangler, the corresponding Atlas integration team, and anybody else you would like to be involved. Generally speaking, in terms of our data release policy, we want to release the data as soon as possible after generation, but we appreciate that in some cases you need to first publish your data. The Atlas Assembly standard operating procedure, you'll hear a bit more about this with Malte Luecken's presentation. This is ongoing, so we're hoping to release it more towards the end of the year. It's been a work of love of many of us coming together. We appreciate that there's not a one-size-fits-all solution when it comes to Atlas Assembly. But at the end of the day, we still need to find a way to agree on overall strategy so that we can generate reproducible atlases. Or at very least, when we release our atlas, anybody can read about how we got there, right? So decide whether you agree or disagree and how we keep on building upon it. And so the SOP that we're releasing is not about giving you a specific step-by-step map of how to do it. It's more about recommendation about key strategic decision that must be made when you build an atlas. And once you're done building the atlas, we want people to release all the details of these different steps and decisions you made, including your code. So the first chapter is about setting up your project, how you assemble the team, defining the scope of the atlas, thinking about this landmark studies we talked about. It goes over more details of this ingestion data, relates to the other documents I just talked about, how you think about data quality, how you perform QC and batch analysis. Again, we're not telling you this is how you do it. We're saying you must do some quality filters, you must do some batch analysis, and please report it in detail. Then we talk about data integration. We talked about how you prepared, where you evaluate the integration. We're not telling you how to evaluate the integration, what metrics to use. We're saying you must evaluate more than one integration method. You must try to optimize your parameters used. You must use some matrix and tell us which matrix you use to decide that this was the right integration method to use. We don't tell you how to define the marker genes population, but we tell us you must define some marker genes population and tell us how you did it and why. And then make a recommendation of how to approach broad community involvement to annotate your atlas and for downstream analysis. So you'll all be made aware of when we release this information, and this is gonna land on the same page where we are releasing all of our documentation about policies and procedures. There's teams that have been hard at work over the last 7 years to make sure that this ecosystem is GDPR compliance. And so I want to talk a bit about this in slightly more detail. Right. So over the last 7 years, some ad hoc task force funders as well as legal groups have been really, really hard at work to make this. Don't take this for granted. This was a lot of work to get there, to empower the global community, to have a system when all of us can share data around the world, to contribute data sets and really achieve a goal of building a diverse atlas. And we're so proud that actually this is now in place. We had a soft launch a couple of months ago, and just in time for the general meeting, we had the whole process approved, and now we have the first data sets out there. So now we can provide end users access to rich metadata with personal information and raw data from virtually anywhere in the world. HCA can store data requiring access, such as FASTQ files. And I want to stress here that this is not for every data sets out there. This is for data sets that we're using to build these gold standard atlases. And it's supported by an international HCA specific data access committee. And this is how it works. You visit our HCA data repository and through the portal. You will search for the data set you'd like to see. And then for managed access data, you'll see a tag here that says access required, and then you click on requested access. And a big shout out to AIDA, the Asian Immune Diversity Atlas, for working hard with us for the soft launch and making the first data sets available on the portal. And please join me too in congratulating the team that has worked for 7 years. I'm clapping on my hand here, because this is like a lot of work to get there. This is a big milestone. You've heard about HCA Extincting. I want to go over this again, especially since we're here in Italy, and some of you may have to deal with GDPR compliance. And so, as Sarah mentioned, HCA is supported by 2 non-profit organizations, HCA Inc., which is in the US, HCA Extincting, which is in the Netherlands, and they work together to ensure compliance with international data protection regulation. HCA Extincting helps European researchers share their data safely and more easily with other jurisdictions. And I want to stress that that's something else that HCA is uniquely positioned as a consortium, in that we are GDPR compliant in terms of regulation. Finally, I want to spend the last few minutes in talking about how we want more global participation. We're going to talk a bit about recruiting Bionetwork champions, expanding project leadership, how Biological Network can help support atlas building, and so give you some example of great community engagement. Right. And so one of the thing we've heard a little bit is, well, how can I contribute? You know, it feels like they're just a small teams that are doing atlas assembly. And I'll take a moment on the side to say, well, actually everybody here needs to participate to atlas assembly. But from a practical standpoint, when we first gather all these data sets and integrate them, we cannot be hundreds of people doing it. So there's a smaller team composed of integration team, Biological Network champions, and of course different members of the data ecosystem working together to do some of this initial groundwork so that we can then engage you more globally. And that's why some of you might be wondering what have we been up to, because we've been a little science. That's what we've been up to. Building this ecosystem, retooling, and starting to do that hard work. But we want more champions. So if you're excited about this, and you're excited about even more than one biological network, you're allowed to be part of more than one biological network and be part of as many biological networks as you wish. Do reach out to your coordinators. If you're not sure how to navigate this, you can go on the HCA website and you'll find all of our contact information. And so we're looking for more champions. And similarly, we're interested in expanding leadership across geography. So regardless where you are in the world, we want to hear from you if you want to take more active roles and if you have new ideas. Remember, we're a grassroots initiative. We're all doing this together. And so John was saying, well, Chloe keeps on having more title. Why is that? It's because I keep on raising my hand. But all of you can raise your hand and help us kind of move along. Actually, I would love to give back some of my titles. So if some of you like to get engaged, that'd be really great. At the end of the day, all of this Atlas endeavor, you saw how we were organized in terms of governance with the different working groups. They're all there around the biological network, right? So our 18 biological network are the core of defining this what are our Atlas assembly effort and where we're going, right? The different 18 biological network are building these roadmaps. And as contributors, you get to shape these roadmap. Do reach out to your biological network coordinators. Let them know how you like to contribute and shape this roadmap. And so, to give a few examples, so I already mentioned that biological network and the coordinators are selecting these different landmark data sets. And there's different engagement activities. I'm going to give a few shout out to a guy who's done a really great job at bringing their bio-network together. They had, for example, in-person 2-day meetings. I think Jose may mention this in his presentation. I love he calls it a mise en place. I'm going to let him tell you more about it to kind of help set up their Atlas assembly. And so I mentioned there's currently a small team that are working for across every biological network to build these data objects around SOP. And of course, all the code of these Atlas assembly effort will be released for sake of transparency. And many of the networks have progressed and published their roadmaps. And this is, again, just the first version of the roadmap. They're going to keep evolving. So do reach out if you want to contribute to the next roadmap. The GAT team meets weekly and they share progress, update about emails after meetings. Development and meet bi-weekly. These are just a few examples. And with phase 3, we're going to talk more about this after the break, where we're going to invite the entire community to get involved and give feedback on the annotation. So you get to all participate on what will be the product of HCA. So there's still so much more to accomplish. Again, you know the paper bundle is coming out, but keep in mind that this is just the beginning of the journey. This is not the end of the journey. V1 is not the end, it is the beginning, and it's an exciting beginning where we're starting to see concretely the product of the Human Cell Atlas endeavor. So in the short term, over the next year and a half, before we get to our 10-year anniversary, yes, it's our 10-year anniversary in 2026, we aim to have finalized version 1 of our Atlas, right? All of us, meaning across all 18 biological network, will have needed to finalize releasing our tier 1 and their tier 2 metadata, tier 2 being extremely important to empower downstream biological query. And on the medium term, we'll have to all engage in developing consensus annotation across these 18 version 1 atlases. And we're going to talk more about this on the next session. And please, there's a lot of people that are there for you if you don't know how to navigate all of this. You saw the face of the executive office. You can find them on the website. There's a cell annotation platform team. I'm going to introduce them on the next talk, the integration team and the data repository team. And you can always find me as well. And we're all there to support this endeavor, which is your endeavor. And this is a team sport. We need all of you to engage to make HCA a success. So with that, I'll conclude, and I'm happy to take a few questions. Thank you. That's very heavy in terms of information. These slides will be available.
That's right. There are microphones circulating. If anybody has a question, just please raise your hand.
Everybody needs coffee. Yeah, Aviv has a question.
So first, before, maybe I should do, well, hopefully you can hear me like this. Before I ask a question, I wanted to say thank you, because I think this was actually a heroic effort made of a bazillion parts and pieces, and they're all combining together, and it's really impressive. Yeah. I wanted to ask maybe more of a clarifying question. How should people think about their engagement with this panoply of activities? Should they think of themselves more as they need to deliver their data in a certain way, or should they think of themselves more as they should come into the Bionetwork and start suggesting it should be, you know, the data sets should be like this, or the annotations should be like that? Like, how does a data generator engage with the data ecosystem? Maybe that's the way I'll ask it.
Thank you, Aviv. That's a really good question. I think you hinted at it. There's like so many different ways you can engage with the community, whether you're a data generator or not. But let's say you're a data generator and you're interested in contributing, please do reach out to your Bionetwork coordinators. That's the first place. We need to know you exist. We need to know you have data sets coming up. And because we're also there to, as the word says, coordinate. We can also identify people around the world that may also be complementary to what's coming up. And we are eager to know what's coming in the pipeline, even if it's 2 years down the road. So do reach out even if the data is not all generated. It also gives us an opportunity to tell you upfront what are the file formats that we're using, so that none of this comes as a surprise. So hopefully, like right now, is of course a bit more challenging because a lot of the data is there. So we need to kind of retrofit some of these schema. But the more you're exposed to it, the more it will become second nature, a little easier. So do reach out in that sense, not only about the data, so that we can, but also in terms of us telling you more about the schema, which is schema values. We're talking about end data objects here that we're standardizing. And also none of these metadata will be such a surprise to you because most of you are already collecting them. So that's one. In terms of which data sets are included for an Atlas assembly, this is part of a broader conversation. And for the most part, if it aligns with the goal of your Bionetwork, they will be included. In terms of engaging, if you have a data set that has already been published, we still want to hear from you. In many cases, we don't have all the metadata. You have an opportunity to reuse the data and get another publication for your trainees out of it, to engage and help in building this metadata, both in terms of the biological metadata, which may require more work and will be recognized through proper authorship, and the metadata. If you also have an opportunity to make sure that the way you annotated the original data set, which is in the landmark data sets, is taken into account as we integrate the Atlas. You get to tell us if you agree or disagree with how it looks like when we integrate. It's another way of engaging. And if you're not a data contributor directly, but are interested in using this data for downstream analysis, or just a domain expert and want to see how this is growing, we also want to hear from you. We're going to talk a bit more about this through the cell annotation platform conversation, but this is tied to, do you agree with what we're building? So there's like different ways of engaging. And you know, this was a bit dry as a presentation, but I think it's really important. And we need, for this to work, like the data ecosystem thing has been really, really hard at work to build this. But this will only be a success if all of you engage with us in making this a success. And the Atlas assembly effort will only go faster if everybody starts embracing these file format and schema.
Over to the stage, right?
It's great to see SOPs for Atlas building and developing transparency standards for. There we go. It's great to see SOPs for Atlas building and transparency standards being brought in, because it's sometimes mystifying how atlases are built at the very minute levels that really make a difference, the choices that you make kind of along the road. But it's also, the tools that we use are kind of not transparent in the first place. So how do you see the first steps to actually being transparent?
So yeah, thank you, Nelson, for your question. I think it's Nelson, right? Yep. Again, I should put my glasses on. Thank you for the question. I forgot to mention it. So when an atlas is approved and lands on our HC data portal, I show you on the far right, you had the Bionetwork coordinators, and I mentioned you would have hyperlinks to a different repository. We're also mandating that the team release the detailed code on GitHub of how they did the Atlas assembly. That's actually part of approval process to increase transparency of how the Atlas was built. Because to your point, we are not using all the same approach to do it, but as long as we're transparent about how we're doing it, that will help support not only transparency, but reproducibility. And so that's part of what we're mandating. There's I cannot see. Yeah, okay. Yeah, you moderate.
Thanks, Chloe. That was great. It's Michaela here, Heart Bionetwork. So just a comment. I think the people that are here definitely engaged. What we have been finding difficulties, and Leonard, Lucia, and Simon, we have our meetings together with Norbert. I think sometimes the difficulty is to engage the people that are not here. And, we reach out, we send one email or two emails, and some will engage into the relatively complex or relatively simple process of importing the data. But it's a little bit hard to get them on board completely. So what can we do? Because I think those are their opinions are really key, because when we start to annotate, when we start to assemble the atlas, the more people we have on board from the community, the more valuable it will be. If it's just a small group of us, and then there are another 10 that don't agree, the atlas won't be used.
Yeah, that's a good question. You're asking about incentives. I certainly don't. I welcome suggestions from everybody here, because it is a hard problem. We certainly face the same problems on the immune side. So 2 thoughts. First, I think one of the hard piece of it is, of course, we just spent a year building this and developing this, and now, people are thinking that this will happen quickly, but we need to socialize this. And so there's this whole socialization process that actually takes time to increase visibility and talks about these standards more broadly until it becomes second nature. And so as part of these wave 1, 2, and 3, of course, these are not second natures because people are only starting to hear about them. Second, you mentioned about incentives. It's especially hard when the data that we're integrating are already published, right? People are saying, why? Well, just take my data. Why should I give you more? So I encourage every biological network to think about different incentives, whether it's talking about authorships with them reaching out, right? I mean, if you're asking them to put more time to reformat data, and if they have to kind of dig in and think about new metadata, that's not free in terms of time. And so explaining that there are trainees engaging will get a second authorship out of it. And depending on the amount of involvement, you can decide as a biological network what you want to do. Also making, and also letting senior authors of this landmark paper know that you want to make sure that their work is well represented and integrated and interpreted. So it is not easy. And I think sometimes these emails we all get one too many emails. And so finding ways of connecting in person through some of the professional meetings. I know as a long bio network met in some of these professional settings, and I think these in-person meetings kind of help as well. And I saw that Aviv on one right side might have some thoughts because I just actually got the mic.
Yeah, I want to add 2 thoughts on this. It's kind of one of these also long tail problems where you can go and chase every person and everything, and that's not useful. So there's a little bit of judgment that one has to apply on what's material to engage and what we just can't. Yeah. You can't have everyone engaged because it's also unmanageable, but also because some people are more enthusiastic than others, right? They want to and not. So there's a little bit of a coalition of the willing, people voting with their feet and so on. So I think there's kind of 3 categories. If you think that some engagement is material, it's really on us to chase. You have to convince people that that's really important and bring them on board. Second, you have to realize what's critical mass, and once you reach it, the rest kind of takes care of itself. If you have enough thought leaders engaged in the annotation, then that will be it. Those who don't want to engage in annotation will not, but they kind of also at some point lose their chance of arguing because everyone else was there and everyone else agreed on something, and it kind of made sense, and that's what people are going to use. That's part of the realism of it. And the third piece is, as Chloe showed, these are actually versions. The perfect is the enemy of the good, meaning if you try to have everything, you will have absolutely nothing. So instead, you need enough, and that becomes the first version. And the way in which atlases are built today, you can add data to them later. Or you can modify the annotation based on additional information. So it has to pass a good bar. That's why one of the constraints is that it has to be blessed by the biological networks, and we do believe we have experts there and so on. But there's no way it will have all inputs from everybody, partly because not everybody wants to provide input, and partly because if that's what we try to do, we will be here in 10 years and we still wouldn't have had even the first version. Yeah. That's roughly how it plays out. I see Martijn is kind of nodding his head. He can share from the long Atlas experiences. I see Sarah might actually also want to say something on
just to kind of quickly add, obviously, agreeing obviously with everything Aviv said, but a lot of effort can be grandfathered, right, in the first versions, because this is coming after the fact. So we, of course, even though there's criteria for approval, we know there'll be a lot of gaps for version 1 because of all of this, and it's totally okay. And Long has used a core data set where they could gather a lot of information, and then you can project some of it. So there's different approach to this as a solution. And just to add a bit to what you said, yes, Sarah.
Yeah, no, I think, Michaela, thanks for raising this issue. I think there'll be a sort of snowball effect, basically, which is, what Aviv and Chloe are trying to say is that we just need to get going, and then there'll be a positive feedback from the community once they see, this is so useful. having this reference. Oh, but I disagree with this. You know, what hopefully will happen is that people will write or annotate or feedback, and then there'll be a kind of iteration approaching like, 100% perfect annotations and objects and data objects and so on and so forth. So I think that's how it will go. So it's just like, let's just go forwards together. You know, we have a cool little team, around cardiac. We can get this going very nicely. And I'm sure it will end up snowballing, becoming a larger, really awesome object.
And to kind of just end on what Aviv said, again, these are version, right? They're going to be timestamped. And we're not going to get it right, and it's okay. As long as we're transparent about, we won't get it perfectly right, I should say. We won't get it wrong. And so as long as it's timestamped and we're transparent about how we got there, we can iterate.
And it's true for the computational analysis, for the annotations, for many things. Computational analysis, there's very vigorous discussions. Should we do this way? Should we do it that way? We won't do perfect first, but it has to be good enough and pass a quality bar, and then the next version will come. People, by the way, are still assembling things.
So I'm going to, because she doesn't have a mic, so I'm just going to repeat what she says. So perfection not be the enemy of the good. It's important to kind of put it out there and in different instances we'll have to iterate. And Sarah said people are still assembling the human genome. It's an ongoing process. And the atlas, the human body is even more complex. So shouldn't be afraid of embracing this versioning aspect.
Okay, this has already been an excellent start to the conversation. I think in the interest of more conversation and more caffeination, we'll break for a break. I do want to give you a preview of the rest of the morning, which is up here on the screen. We'll return here at 11:10. We'll have the second part of this session, which will focus on cell type annotation, and then a session updating you on biological networks and actually continuing exactly this conversation about how we're engaging with people both within and externally to the biological network. So again, come back at 11:10. Please visit the booths. Please say hi to Michael Fletcher, and please be back here at 11:10. Thank you.
We're starting again. Everybody, let's come in so that we have more than 15 minutes for lunch. We have a packed schedule, so many exciting things to discuss. You're so lucky you get to hang out with me somewhat a little longer. Okay. So we're going to continue the conversation about the toolbox we've been expanding for HCA. And before we start, I don't mean to embarrass anybody, but for the members of the HCA data ecosystem, I don't know who we have from the cell by gene team, cell annotation team, the data repository, and the portal. If you want to raise your hand, I saw Dave, Roger. I know Evan is somewhere. I'm going to force you to stand up at the end. Well, at least you have Dave. Dave is here if you have questions about the portal. A lot of them are here at the meeting. They might be still outside. Do come and meet and ask questions. Okay, so we're going to talk about the cell annotation platform. As John mentioned, I wear many hats. I'm also the principal investigator of the cell annotation platform. We talked about version 1 criteria in the previous presentation and what needs to be done to get your atlas certified for version 1. But for version 2, you actually need, we as a community, and we as a Bionetwork need to engage with the community to derive consensus annotation. That is part of the version 2 criteria. There's other criteria, but this is what we're going to focus on. So how do we get to consensus annotation? And so we talked a bit about this ecosystem, right? You can ask, well, I have my gene expression matrices, my end data object, contributed my tier 1 metadata, what's next? I want to talk about the cell annotation platform. It's not only because you need it for version 1 criteria, you want to contribute to this community, but it's also because it's what you need to set up a cell annotation jamboree. What are the ingredients for a cell annotation jamboree? Well, you need your integrated object with your tier 1 metadata. Nelson asked me about, how do we make sure that we understand how the data was integrated, and so that's reproducible. Like, that's one way about it. You need to know the source data sets. You need to load the data on CAP. We're going to go through this. And you need to have the metadata for individual source data sets. So I want to talk about the annotation metadata goals, right? So we need to consistently and accurately work with cell annotation when constructing an atlas. We need to figure out how we agree on cell identity. It's important to empower quality controls on individual data sets before integration, and also to generate de novo annotation pre-integration. It's important to enable QCing the integrated output. Can we figure out if cell subset commonly identified across several independent data sets clustered together? And of course, it's important to provide the foundation to build consensus annotation framework and figure out the synonyms of different cell identity. But this is actually challenging. Why is it so challenging? That's because a lot of the single cell annotation to this date has been done manually. And that's time-consuming. It's not really scalable. Some of the automated annotation framework rely on what's currently out there, and it's not at scale enough for this to become a gold standard just yet. Very often, we differ in our definition of cell type. We differ in the molecular signature we use. We differ in the vocabulary. We differ in the terms and the label we use for the same tissue, and sometimes for the same cell. We also differ in the precision we use to define these terms. And this is not to nitpick, just for an example, but on the far left, in the red circles, you have examples of two different data sets. In red, where you have very precise terms within a data set and very broad terms. So for example, here you have all immune cells, T, B, and K. I picked them because I focus on immune cells, all together, right? Obviously, they should be split. And so this is not very uniform. And so the question is, how can we systematically and more objectively compare cell annotation provided by different domain experts to derive consensus? And this is how we came to create the Cell Annotation Platform, or CAP for shorthand. It is a community-driven platform to create, explore, and store annotation. We provide infrastructure to accumulate, share, analyze annotation terms with associated molecular signatures to interpret cellular identities. With time, this is also going to be a platform that will provide automated annotation services and have tools to encourage researchers to reach consensus annotation. To make this happen, we've defined 12 annotation metadata fields. Annotation is a very subjective matter, but we can make it slightly more objective by defining specific fields. So there's the names that you're giving them, but more importantly, the rationale. Why did you call a cell a certain way? Did you use unbiased method? Did you use legacy markers that have been used in the literature? Are there existing cell ontology term? If not, what is the closest term? Because one of the things we need to do is also update our cell ontology. We need to regard the synonyms of the same cell type and so forth. And this is the necessary context that is required to consistently and accurately work with cell annotation when constructing atlases. This metadata can then, if we all use the same metadata, and which is very well defined in our documentation, suddenly we are tooled to more objectively compare and contrast annotation within and across data sets. And all these fields are provided by researchers, and we have data curators on CAP that can go through all of them. I want to shout out to the Retina Bionetwork. Their atlas was approved, and they actually the first one to have all of their objects annotated on CAP. And they've been really critical in giving us feedback and making sure this is a user-friendly process. So a very, very big thank you to the Retina Bionetwork, and we look forward to working with the next Bionetwork. And obviously, it's not one at a time. We're eager to work with all of you. And so there's many features of the Annotation Platform. I won't go through all of them. We have Evan, who's here, who's our lead scientist, and we are eager to set up user interviews. But it's important to note that we have workspace that is private. So even if you have unpublished data sets, it's like Google Docs, you can work in private space, invite people to annotate your data, and you publish when you're ready. We have browsers where you can either navigate pre-computed differential gene expression and embedding, but you can also do calculate live new differential expression values. With time, we're going to also be releasing automated annotations. You can look at relationships between annotations, and with time, we'll be able to download these cell cards where we're going to have some of the first consensus annotation coming out. So, this is a bit dry, so I wanted to show you some videos to give you a sense. So this is the landing page, and so, this is working. So you can go see in the data set, so you have examples of data sets that are here, and you pick one data set, you get information about this data set, the different cells that are on this data sets here. You can click on the study. Within the study, you get the full list of the different cells that were in there with the number of cells that are present in each of these data sets here on the main. You can go in the relationship between these cells and other data set here that you're seeing across the different cells, and this is a long, I think it's a long data set. And then at the very top, you can go and click on the molecular data tab for that particular data set. Now it's going to load the embedding with all the cells, and the genes are differentially expressed. In this case, this data set was already annotated. And so on the, you can see on the far right, for every cell subset, the clusters present, you have the different marker genes that were defined by the contributors that are pinned, and you have them all. So you can look at all of them by expanding here on the far right. You can, go back to the embedding. You can look at specific genes by pinning them, and this is, we're looking at PPAR gamma. It's going to show you that they're mostly expressing in these alveolar macrophage, among others. And as you over, you can see the different differential expression values. These are obviously snapshots to give you a sense. You can go back, you could re-annotate, you can go back to the author cell type annotation and kind of zoom out if you want to. This is just one example. You could have a new data set that you want to annotate. And so here is the same data set. I'm zooming here. You can use the polyhexagon tool to kind of select cells where you want to. This is live computing, right? So it's not always pre-calculated, so you can adjust your notation. So we selected these cells. And now I want to go and I want to do differential gene expression. I can do it against the background or against customized selection. Here it's showing me the genes that are upregulated in this red cluster compared to the rest, showing the genes that are more highly expressed. And as you hover over, it gives you a summary of the function tied to these genes. So you can decide, I want to pin this particular genes to have it as a marker, right? And I check, and it's very much more uniquely expressed in this particular cluster. And again, we're hovering over, so you can look at the expression level here, and it's showing you other genes that are more uniquely expressed compared to the rest, where you see it's not expressed in other cells. So I feel pretty confident this is unique. So I want to go and create a label set. I'm going to give it a name. So obviously, we're making this up for the sake of this demonstration. We're adding it. And then you get a window that are going to open here, where you're going to create a label, right? And now you're going to start adding all of your metadata. And you get prompts, which is meant to kind of help us derive more standardized nomenclature. So you enter all of this information, and when you're done, you submit. And for sake of time, I'm going to keep on going, but then you would, say, add your label. And once you add your label, it's going to be added on the embedding. See, now you have a label here. So this is your annotated de novo. You may also have a data set that is already annotated by colleagues, for example, and you wanna refine the annotation. That's another example that's already annotated. I'm selecting these cells here. It's telling me which genes are more uniquely expressed. Again, it's telling me something about the biology of these genes. It's showing that the sets of genes are more uniquely expressed, but I'm questioning the fact that this little cluster next to it may look actually very similar. I'm actually looking at the top gene, which is also expressed on the cells next to it. And of course, this is just for sake of demonstration. But I'm going to conclude that these are likely the same cells. So to check this, I'm going to select these cells compared to these cells, the blue versus the red, calculate differential gene expression live, and realizing that, they all express the same gene. So I think these are the same cells. And so based on that, again, you can hover and look at the different gene expression levels. So based on that, I'm going to correct my label. I'm going to go select these cells and call them the same way. I'm going to add a label data set, create add, and then I can select groups of cells to be add the label, and it can complete my annotation metadata. And so for sake of time, I'm going to skip this, but again, you get prompts as you kind of complete the different files. So you can imagine that if all of us collect systematically these metadata fields, we now have some material to start comparing and contrasting, right, these annotations, because these annotations remain opinions. That means we can start getting feedback. We can do rounds of vote and start addressing the disagreement. And Mikhail asked me about, we talked about versioning before. Well, this is one way of starting to version of our annotation. And so we also released a couple of weeks ago our feedback features. They're in production and want to hear actually your feedback on our feedback features to see what works and what doesn't work. So you can say, you can go over some of the annotation, for example, of the integrated object, and you could say, thumbs up, I actually agree, I approve, no further discussion, what does it look like? And so this is from the GUD Atlas group. This is actually one of their data object. Is this going? So these are the different cells they have defined here. And you can click. So I'm going to pick neuroendocrine. You can pick on them. And you can say, well, I'm looking at this particular marker. I'm selecting these. I want to provide feedback. And in this case, I'm saying I'm agreeing. And I'm going to write a justification for why I agree. You don't have to write a justification for why you agree, but you can also write a justification for why you agree. This looks right. I use the same markers. You know, and then when you're done, you can say, send your feedback, and we compile that information. And then you could also say, well, I agree in terms of its molecular definition. I agree that these cells should be together, but actually disagree with the metadata you use to define them. You know, I would have used slightly different marker genes. I disagree with the way you define the ontology. And so how does it work? This is not a video, this is a screenshot. And back to the neuroendocrines. What I could have done is actually I could click on refine metadata, and then you get windows open for each, and you can say what you would suggest in terms of refining the metadata, and then you can click submit. And then this is all compiled. Beyond refining and agreeing, you can obviously disagree. You can say, I actually disagree. These groups of cells should either be merged or should be split. Right, so you can, in both cases, whether you merge or split, you need to give us on CAP a justification. The number of clusters, the markers, and why you're getting there using the metadata. So this is, again, an example. Again, this is just meant an example. So the T cells, as you can see, was a big cluster out there, right? These markers are very broad. And so we're selecting these cells, we're showing these are the markers, we're selecting these cells, select, and see, you're gonna provide feedback. You can disagree, split, or merge. I'm gonna say split. I'm gonna say, actually, we want 3 groups. Again, we're making this up. Don't take it for cash. Those of you that are immunologists, it's just meant an example. And it's saying, well, the first name should be this. These should be the markers. Should be the second name. This should be the markers. And at the end, you give us a justification for, you know, why you think this should be considered this way. And this is, then when you're done, you send your feedback. And so at the end, you know, we can open it as a Bionetwork for a certain period of time. For every cells, you have the number of cells, you get the number of people that give, you know, agreement, disagreement, and comments. So we get feedback summary for every cell label, you get a summary for every embedding. And it's important that we record all cell annotation and authors. It's transparent public. The reviews are transparent and public, and this resource will remain publicly available as we version it. And this is our first framework to help us work towards consensus annotation. The consensus is important in defining the groups of cells we agree with the whole metadata schema. It is conceivable that many of us are looking at the same cells and calling them different names, but once we start doing this exercise, we may realize that at the end of the day, we're just building a dictionary of synonyms, and that would be incredibly powerful to just accomplish that. Working with this, it will also give us a better gold standard data sets to generate automated annotation frameworks that are more reliable. And so that will be one of our outputs. Importantly, that will help us update the cell ontology, working with the cell ontology teams. I know David Summer and his team are in the audience, definitely connect with them. And as importantly, it will help us figure out where we disagree. Figuring where we disagree will help us, our community, focus on spending more time in solving these disagreements and resolving these conflicts. And so I mentioned this documentation earlier on in terms of a document that defines all of the different metadata schema, including CAP, highlighted in yellow. The website is live. We have an amazing team ready to help you us in terms of like doing user interviews and getting you onboarded there. We're not going to talk a lot about Jamboree today. We're going to follow up with more details in months to come. But we're there, the CAP team is there to help you tailor CAP demos for your Bionetwork. We're gonna, once you're done with your version 1 Atlas and have all of the data and data ecosystem, working with the integration team, the executive office, the CAP team, will help you think about the scope and format of your Jamboree, whether it's in person or virtual, depending on, you should definitely have hybrid so that everybody has access to this effort. You need to think about your desired output, how you want to engage, what you hope to achieve. Help us help you. We're there to help you. That's what we're here for. And so, Evans, wave. I'm going to embarrass you. Evan is far back at the right. This is Evan. He's here for the whole meeting. Go and meet Evan. Mary is also our amazing data curator, and they're there to help you. So with that, I want to say a special thank to Yingli, Rach, and thank you so much again to the Retina Bionetwork for all your help. Felix Fisher I didn't mention, and talented graduate students who developed new automated annotation frameworks, to Lucia, Ellen, the executive office team, and so many others. Thank you so much for all your help. This work was served by Peter Karchenko, who was leading this annotation platform. It's been my greatest honor to kind of help continue the work together with Niels Gellingsberg and David, who are also in the audience somewhere, I think. Hey, Niels and David, they're there. Also connect with them, also to Fabian, Aviv, John, and Peter for all the support over the years. This is an amazing CAP team, and a huge shout out here to my right to Evan, to my left to Evan, who's really been pushing through this to the finishing line. So in terms of the next talk, we want you to hear from both the Gut Bionetwork and the Brain Bionetwork in terms of how they're approaching cell annotation and what are their plans to use in CAP. So I think next we have Jose. You're here. Jose, you're next. And so Jose is going to tell you a bit more about Atlas Assembly with the gut and their plans for using CAP.
Thank you, Chloe. And just want to take an opportunity as we get set up here to thank the organizers for getting us all here together in this beautiful venue. I know it's not easy to pull these things off. So as Chloe mentioned, what I wanted to do is take a few minutes to walk you through this approach and how we've developed a training plan for our gut integration group. And so I'm just here presenting the work on behalf of the HCA gut working group collectively. So these are the two team leads. None of this would have been possible without Chris Lance, who's a PhD student, and Kyle Kimmler, who's a senior open data scientist at the Cell Discovery Network initiative at Boston Children's Hospital. So where did we start? This actually started, in the genesis of this discussion started off at the HCA Vienna general meeting in 2022. There was a nice breakout discussion facilitated by Matthias Zilbauer in participation, I'm sure, with many of you here in the audience, where we put together this general roadmap for the Human Gut Cell Atlas. And this is really, I think, quite a nice document that summarizes a lot of our thoughts. And one of the key points is really that the gut isn't just one organ, right? It's many different organs that are connected to each other. But if you talk to a specialist in the gut, the duodenum, the ileum, the colon, they have nothing to do with each other, let alone some of the more specialized tissues, right? So it's really something that needs to be paid attention to. And the other thing, of course, is that it's highly reactive to its environment. What all of you may eat at home is not the same as what you might find here in Milan. And so depending on when or where we sample the gut, this is going to be very different, of course. Now, fast forward to 2023, in a meeting facilitated by the Helmsley Gut Cell Atlas Consortium, we had a couple of different sessions on this. So again, there was an annotation integration session, and there was a breakout session. But building an atlas doesn't just happen in an hour, right? And so this was another kind of set of discussions, but we didn't have that real kind of beautiful framework that has been very carefully thought and laid out by a lot of the individuals that Chloe and Aviv and Sarah helped introduce this morning. And so what we've been able to do is really, in close collaboration with Chloe and a lot of the individuals that she introduced, is over the past year, since that meeting, actually go forward and say, here's the data sets, here's the basic building blocks that we have to operate from, and then move forward with the harmonization of metadata, data integration validation, our Bionetwork meetup, which I'll discuss a little bit more, and now getting towards the tail end of 2024. And so you've seen this slide before, I think in horizontal and vertical formats, so I won't belabor the point, but this is the general data structure as how we get towards Atlas assembly, and finally these things coexisting together in the HCA data portal. So when will version 1 be complete? We've also seen a few versions of this slide. What I will do is focus on the integration and batch correction part and the tight link with the cell annotation part and of course the CAP structure. And this is really where the community involvement is a key piece. And so Chloe alluded to this. In discussions with her, I decided that, what do we actually need to do to get towards this atlas, right? And I said, let's have a mise en place meeting. And I introduced this concept to Evan and to Chloe, and here I'm showing you a nice picture of a paella, right? But what goes into making this is a lot of different ingredients, right? So even though it's a beautiful blend of different things together, what we really needed to do is to understand how to deconstruct it in order to make it more of a community-driven process. And so, again, supported by a generous contribution of Helmsley, what we were able to do is convene together a meeting in June in Cambridge, UK, that was our mise en place meeting, right? And I emailed out a few people. Got Bionetwork, got Atlas champions, Helmsley GCA grantees, large data set contributors, people who I know are in this room as well. And the main goals really were to lay out our different pieces that we needed towards getting to actually building the Atlas, right? And so these were the individuals that participated in this meeting. And out of these individuals, right, first 2 days all going really well, then the third morning comes around and you say, okay guys, it's not just about what we talked about in this room, right? When we actually leave this room, we need to continue being engaged, right? And I think that this is oftentimes where these discussions fall apart, right? And so in order to do this, what we did is we leveraged the fact that we happen to have, I think, a lot of runners. So Chris and Kyle met and bonded over their shared love of running. Our program coordinator at Boston Children's Hospital is actually a coach, and so we set up a training plan, right? And so he said, this is, okay, you have your target event, right? How do you work backwards and actually build a training plan? And so what we said is before leaving the meeting, we said there's going to be an M group for metadata, an IA group for integration annotation, a C group for use cases. And we said there's going to be meetings weekly, Monday, Tuesday, Wednesday. If you want to join these meetings and you're not currently a part of them, please email either of the email addresses listed down below. And so what these meetings we're basically tasked with doing is not just to have more Zoom meetings on our calendar, but to actually move these things forward. And so the metadata collection group I'll just share one kind of brief vignette in what we've been able to do there, and I'll share a bit of the approach of how we've been taking advantage of the integration annotation working group as well. And so metadata, right? This is something that I think strikes fear in a lot of the scientists that are working. You say, oh, great, I have my paper, it's nice and pretty, right? I get an email in my inbox saying, please contribute your additional metadata. Like, oh, that sounds like a lot of work, doesn't it, right? So, what can we do to make this a little bit easier, right? How can we try to incentivize this? So Kyle, in collaboration with the working group, has developed this pipeline here, where we actually start to pre-populate a metadata sheet for you. So you're not just staring at a blank Excel sheet or Google sheet in this case, but you actually have existing data we've been able to scrape from the paper and start to pre-populate for you. We then share this with the authors, and then we share on top of that a correctness and completeness graph with all the different data sets. So you can kind of see how you're doing in comparison to others. And then we start to update those graphs as we get more and more information coming in, right? This, of course, is not perfect, right? We need to better incentivize and have a nice structure for how we can appropriately have everyone here that is participating in this feel that they're really contributing and really make sure that we have all the critical pieces of metadata together. And so why do we need all of this metadata? Well, one of the things is that, and I'll just show this here while I get to the next slide in case anyone wants to take this down. This is now available on GitHub as well. So why do we need this metadata, right? The point of the metadata, maybe naively at first, I thought was, well, let's just correct away different pieces, right? Sequencing batch, where things were collected, et cetera. But there's actually more to the metadata that we need, right? What we need to do is not just correct these technical differences, but really to understand them. And I hope that Malte also, as one of the integration leads, will help to be able to shed a little bit more light on this in a subsequent talk. And so this is really, really key piece of information. We need to understand what's confounded with each other and what we can analyze discretely. And so in terms of integration annotation, and this gets back to tie into what Chloe was sharing in terms of the cell annotation platform, what we've been able to do is pull together a whole bunch of different studies. These were deemed high quality studies in the literature. So this is why you're not seeing necessarily the, through the roof cell number, but we've really worked on identifying studies with high quality data and metadata available to us. And what we started to do is compile different high-level pieces of metadata. Now, I introduce, of course, that the gut is this connected tissue that goes along the length of the organism, but there's also distinct layers, right? And we all know that people may have sampled distinct layers in the gut as well. So this is one key piece of metadata that we need to understand. Are we looking at just the epithelium, the lamina propria, the submucosa, how deep into it? How does the cell and the nuclei and the type of dissociation influence all of this? And so these aren't things, again, necessarily just to correct for, but to understand so that we can propose how to better collect the next version of data that will go into a subsequent atlas. And so really one of the key lessons, and this is probably the main take home and why I think that the CAP infrastructure is so critical, is that we can't just integrate over and understand how good or bad our integration was if we're just dealing in cluster numbers, right? We need to understand what the accurate annotations are that underlie this in order to have our accurate understanding of integration. And so what we did was we got together and extending on conversations from that initial meeting, we were able to define the cell type hierarchy. And I'll get into why I think that a type of hierarchy or taxonomy is a key way of interpreting this. So we came up with about 163 different cell clusters. Again, these are organized along the hierarchy and markers for each. And this ends up being some of the initial input that we put into the CAP ecosystem for some of the studies as well. And so this is why I think it's really important. And this is the importance of growing a tree from a strong foundation, right? So I think we can all agree up to a certain point, right, about certain cell type annotations, right? What's a T cell, what's a B cell, things of this nature. But then if I'm talking about dendritic cells, and I'm talking about dendritic cells with Chloe, then I know that if I just talk about dendritic cells, that she's going to be, well, what type of dendritic cell? And did you look for this marker? And oh, but what about, and there's a intense level of detail there that's important to understand, right? But if I'm talking with dendritic cells with Malte, and he's an expert in a different cell type, right? He might just be like, well, great, dendritic cells, we clustered to completeness, right? So we need to know where we are along the branches of this tree, right? To really be able to define this. And so here's a nice example of, CDC1s. Maybe Chloe understands that there's actually more diversity here, but probably for everyone here in this audience, we can kind of agree on the general substantiation of this definition. So what are the central questions for each group? In, I think, the metadata group, and I'm happy to receive kind of additional questions and feedback here on these points. We want to understand how to incentivize metadata, and particularly cellular metadata contribution. For the integration annotation group, do we enforce a known cell ontogeny, or do we really let the data speak to us, right? And what are the benefits? Maybe we need to do both. And for the C group, which I didn't get a chance to get into, the use cases, how do we kind of blend this frequency and gene level data to effectively map patients eventually onto a reference atlas? So this is kind of where we hope to go. Now, just in the last 20 seconds here, I think that everyone here is probably interested in single-cell biology to some extent. I have the great privilege of helping to organize the next single-cell biology meeting in Whistler in May of 2025. I guess the Keystone Symposium was well aligned with the organizers of this, because in parallel, we also have the Cells in Context organoids meeting happening at the same place at the same time. So I hope to see many of you there. So with that, I just want to acknowledge the core team that helped make all of this possible. coordinators, champions, HCA members, and so thank you for your time and attention, and also my lab, so the main day job at home. So thanks for that, and I don't know if there's time for questions now or if we do discussion after. Perfect.
Hi, everyone. Is this working? Good, okay. Hi, everyone. I'm Nelson Johansen, a scientist from the Allen Institute. And I wanted to share some of our work on annotation in the brain as a part of the Brain Initiative Cell Atlas Network, BICAN, and how we can utilize what HCA is doing and how CAP is working on enabling community annotation efforts. So a quick overview of the Brain Initiative Cell Atlas Network. This is a large consortium of groups looking to build the next reference atlas of the human and non-human primate brains. So a lot of diverse expertise, from epigenetics to studies of variation and cross-species comparative work, as well as the actual effort of building these foundational frameworks or references in human, macaque, and marmoset whole brain that Edeline's UM1 is driving forward. And this effort is looking to build these references that can be used by the disease communities, the variation communities, to align their work to and have these common nomenclatures to discuss cell types. So a bit more about Edeline's UM1, which we've called HMBA, the Human and Mammalian Brain Atlas. This is a functionally guided brain cell atlas. And what I mean by that is that we've done fMRI of cortical regions to map functional aspects of the cortex across the macaques, marmosets, and hopefully human. So we can very carefully dissect the same functional regions across individuals, as well as, of course, profiling the rest of the brain subcortically. We're doing this with 10x multiome, RNA sequencing and ATAC-seq, spatial MERScope, as well as xenium, and patch-seq profiling, so the morphology and firing patterns of individual neurons. You may be aware the SLEDI Atlas was recently released, which is a great foundation for us to build off of. 100 different dissections, 3 million nuclei. HMBA is aiming for all subcortical structures with the fMRI mapping in cortex, and totaling around 4 million nuclei across all 3 species, 40 million nuclei across all 3 species. And some of the key take-homes from this work is going to be these atlases, work across species, and the multimodal aspect of how you define cell types, and how do you integrate all that together into a consistent nomenclature scheme. So, of course, we're building off the backs of giants here with the mouse whole brain work. My colleague Zhizhen Yao, they produced this amazing reference atlas of the mouse whole brain. 5,000 clusters, 7 million cells and nuclei, with spatial transcriptomics that does an amazing job delineating these clusters in the mouse brain, which is really striking to see. And of course, on the human slide, the SLEDI reference just talked about gives us a nice foundation to build off. But there was, it wasn't full coverage of the whole brain. There was a limited number of dissections, but really the landscape has been set by SLEDI et al. So we're trying to start simple in BICAN, and that's focusing on the basal ganglia, which may not be a simple structure, given its associations with limbic, motor, and associative circuits, talking throughout the brain, as you can see in these connectivity diagrams. And we're profiling, in basal ganglia, all these core regions involved in these functional loops, because they're associated with different diseases. Parkinson's being a striking one that we're really interested in. So, basal ganglia, or BG as I'll refer to it, is a core set of structures: caudate, putamen, nucleus accumbens, globus pallidus, And really interesting organization. The caudate, head, body, tail kind of wraps around the brain, and striking diversity of molecular types in this area that we're looking to cover across all these different BG regions. So kind of the question to us is, these atlases keep popping up. Every single cell study has their own way to name a cell type. And we're hoping to kind of break that cycle here with a truly consensus nomenclature. And Ed Lean likes to refer to this as the similar to the Human Genome Project, where we build a reference with the community involved, and this gets then mapped to by others in the field to associate their work to this beautiful consensus nomenclature. Consensus nomenclature for us means what we've been talking about this morning, but it also means a species angle as well. So we're looking to build nomenclature that's conserved across multiple species at once, which it's a hard task to take on, but we think we can make some progress here. So, consensus requires that we get the community involved, and we're also aware of the community and the field as a whole as we build out this nomenclature. So I like to think of this as a lineage, essentially. HMBA work, we're building off of all these previous studies and more that have defined nomenclature about the basal ganglia. But what this ends up creating across these studies is a kind of slew of different nomenclature for these cell types in BG. I'm showing here these direct pathway medium spiny neurons, and I'm showing you 7 different names for saying the exact same thing. And these have been studied for decades, but we all can't agree on a common terminology. So with the HMBA work, we're looking to finally align on a common name, get the community involved in agreeing on that, and then moving forward with just this name, and people can then map to this annotation and know we're talking about the same thing. So of course, what I'm talking about today is just the first version of this basal ganglia consensus annotation. As we look whole brain, our view on cell types might change. There can be similar types to MSNs in different regions of the brain that you want to capture carefully. So this is a versioned annotation process that we're just getting going. So we also think about cell types as a hierarchy at the Allen Institute and these BICAN projects. So on your left here, this is a taxonomy view of our current consensus work. Neighborhood, class, subclass, and group are how we organize cells from very coarse to more granular nomenclature, showing you the number of clusters for human and macaque, number of cells, regional distributions of these cell types. And then on the far right of this figure, the names that we've used. And one thing worth noting is that there's a little book icon by every name that has support in literature. And the work that we've done has been to embed these common names used in the basal ganglia field into our nomenclature, but also look for conserved marker genes that'll work if you're looking at mouse, human, macaque, which adds a layer of complexity, but hasn't really been done well to date. On the right, again, linking out to all these previous studies, I'm showing an integration of HMBA along with the previous single cell work in basal ganglia. And the take-home message is that there's a lot of conservation of this these cell type structure across studies and species. So looking, to also compare against what's been done, we've been linking our nomenclature system, which is on the right here, back into the mouse Holbein work from Yao et al. And there's a lot of one-to-one homologies that we can capture. And these are pieces of evidence that we need to keep track of and communicate because it allows the mouse field to kind of understand their typing in our new, hopefully, consensus context and move forward as a field. Showing here MSNs in blue, but also, and many of these are conserved across species, but there are types that we find arise uniquely in primates, these TAC3 interneurons, which we also have to be able to capture and encode. So I talked about, to us, consensus means, community adoption, but also multi-species alignment. So looking at genes is not just one species. Here are two GPI core and GPI shell types that are involved in different connectivity patterns in the basal ganglia. On the bottom, I'm showing multi-species views of 3 genes. TBR1 is a deeply conserved marker for GPI shell. Great gene to use in a name. SST and PVALB, they're divergent across species, so you wouldn't want to use this gene in a nomenclature system that is in consensus. And we cluster single cell data, that's our basis for cell typing, but really it's the multiple lines of supporting evidence that allows us to take a cluster and call it a cell type. So we're profiling with spatial transcriptomics that tells you where clusters organize in the brain, patch-seq morphology and firing patterns, and also viral tooling. So how can you use the genome, the epigenome, to target individual cell types with viral tools? And all of this builds evidence for why a cluster should be named something. And capturing that is still a difficult problem for us. So just an example of one of these modalities, here I'm showing a spatial profiling of the striatum, caudate putamen nucleus accumbens, and painting on for every cell our consensus nomenclature. And you can see this beautiful organization of types. These striosome patch MSNs, medium spiny neurons, organize in these little patch structures of the dorsal striatum. And then you have unique cell types that arise only in the ventral striatum, as well as olfactory tubercle. So this gives us a really nice evidence to describe cell types and clusters. And we've been starting to use CAP to help us understand how these cell types organize on the single nucleus side, and trying to link these two modalities together. So this all builds, these differences build a foundation for us to then do more work. So my colleagues in the BICAN are looking to understand diversity across humans, across primates, and in disease with the CAD project at the Allen Institute. And hopefully, the goal is that these studies will all map into the same consensus system, and we can all talk about the same cell types in a common way and do joint analysis. So tools to enable these nomenclatures there's a lot of them, but we're still trying to figure out what is the right approach for us. How can we bring in multiple lines of evidence and encode that carefully so that machine learning, like large language models, can capture it and help us communicate this nomenclature. LLMs provide a very nice framework for us to populate fields from literature and also communicate to various levels of audience, from high school to college to experts, these different cell types and their information. So CAP is an exciting opportunity for us to work as a community in BICAN, as the broader community, to support annotation. There's also tools from David Sutherland's group on building an annotation in a community fashion. And of course, data sharing tools, Cell by Gene, CiteExplore, they allow us to communicate data with the community as well. So trying to find the right, way to utilize all these tools is an interesting problem for us. So with that, I want to thank all of these people at the Institute, and even more broadly in the BICAN Consortium. It's a huge group effort, and definitely exciting to see these references starting to be pulled together. Thank you.
On time, but we'll take a few questions for anybody here. And I don't know who has a mic, who's running with a mic. Hi. There's a mic there.
Thanks a lot. Really, really nice talks. I'm curious for the last talk. When is a comparison across species? When is there enough evidence to call to clusters or cell types by the same name? Because you mentioned you kind of amass evidence and stuff, but I'm imagining there will be a distribution of similarity.
Yeah, usually we start with an alignment. So making sure that the transcriptomics agrees between two species at the cluster level. But that's the very first link that we have. Then we have spatial profiling across species. So you ask the question, do these cells organize spatially in the same way? That's a piece of evidence to support a homology. And then even more deeply, morphologically and physiologically, if the neurons fire in the same way and their structure is the same, that gives us more evidence that there is a link across species. So it's kind of a tiered system where alignment's the first step, and then we build evidence to, assure ourselves that this really is the same type across species. And as you have more species, it gets a little easier as you build more support.
And I just want to, I was very excited to hear, I got to meet Nelson in other contexts. I was like, Nelson, you must come and speak because, of course, by the name human, we're focusing on human cell atlas, the human cell atlas. But at the end of the day, we also want to be able to translate our findings to do follow-up mechanistic studies, over the next decade. That requires us figuring out how we align with other species. We talked, there's an organoid workshop, but we want to go beyond the boundaries of organoids. And so thinking about annotation through the lens of species and seeing how they're conserved is really critical if you want to be able to accomplish this mission. And this is a good example of, I mentioned during the CAP talk, we should think about other features we want to include when you're thinking about annotation. And thinking across species is an excellent example of the type of feedback we want to hear. And so, yeah, that was great. Do you have any other questions? I see a question.
A follow-up on what
yes.
Nelson just said. So you mentioned the comparison. You said we look if they're spatially the same. Can you say a little bit more about how you actually compare at the spatial level? I think in the RNA or the molecular profile level more generally, people developed approaches to compare, which still have challenges. But in the spatial level, I'm actually really curious on how you do the comparison.
Yeah, my colleagues in the spatial group, they use tools like spatial domain detection to organize the MERS-CoV experiment into these, analogous to clusters, they call them domains. These are cells that have similar expression profiles and similar spatial profiles. And you can do that across species. So then you can match up domains spatially across species. An example, and honestly, you can also see, if you know the brain region well, the expected patterns that have been talked about in literature from stains and other like ISH studies. An example is the islands of Caia and the olfactory tubercle. These are very ventral, granular cells that have been described in literature for quite a while, and they organize into very specific little islands of the ventral striatum, and you can pick that out pretty easily by eye from MERS-CoV experiments, which isn't quantitative, but it's the same spatial pattern occurring across multiple species that gives support for like those homology links.
Well, there's a lot of questions, so let's see how we can get. I see one, I see waving left, so 3 more, and then I think we can take more during the lunch. Yes, go ahead.
Hi, great talk. I wanted to ask if you have some idea of implementing a rating system for different model systems for a certain cell type. So basically, if you could say, okay, I mean, you're saying you have this hierarchical system, but is there some kind of idea to bring that to, okay, if you want to study this cell type, this model system actually represents this in the best way?
Oh, like trying to find the most representative cell species for your work. Yeah, that's a great question. We haven't explicitly done that, but for us, when we do cell type mapping, usually you don't have the appropriate species. Someone who's working on armadillo doesn't have an armadillo reference to go to, but what is the closest species to an armadillo? Looking at phylogenies, we can take our best guess at what species would have the most similar homologous cell types or transcriptomics. So trying to build a rating system on what is the appropriate reference or species, it's a good question, it's a great idea. We haven't thought too deeply, so I'm happy to talk more about it.
I think there was like a lot of waving this way, and then we'll come back to you, Chen.
I agree, those were great talks. Just sort of following up on Malte's, I think that the BICCN really led the way in thinking hierarchically about cell classification and really having a multi-tiered classification system that we can work with. And I'm and now I see your new nomenclature and classification system, and it's like, what happened to excitatory and inhibitory and non-conforming dopaminergic? I mean, just get, and interneurons. Don't forget about those upper levels of classification, and that get us grounded into that in your hierarchy. So, even looking at the neuron versus non-neuron signature, that, for a given population, that's an extremely important signature with which these, subtype differentiated neurons has to network interact in order to determine the biology of that cell. So we've got to have that ability to quickly get onto those upper level classes.
I didn't mean to interrupt you. We just have a few minutes. So I think that's a really, really important comment. Let's not reinvent the wheel. Let's make sure we respect legacy knowledge. And sorry, I'm jumping in, but I think this is also why, while in some of the metadata schema we have on the Annotation platform, we do ask about legacy knowledge. What's in the literature? How does it relate to what is known? Because we should not reinvent the wheel, and we should definitely anchor it. That doesn't only apply to brain. That applies across all organ system. I'm sorry, Bruce, I just want to jump in since I'm seeing the timer. But did you have a question, or is it mostly a comment? Okay.
So we can talk more at lunch, but I completely agree with you. If we don't have the names you're looking for in the nomenclature system, then how do you get aligned with our work? And that's a complex problem, what name to actually choose at the higher levels to communicate these. I mean, excitatory and inhibitory are obvious, right? But we're hoping that large language models can be an entry point as well, a chat bot where you can say, I want to find excitatory neurons. What the heck did we name them in this new system? And give you an easy, low barrier entry to understand multiple nomenclature systems at once.
That's great. Sham.
Really impressed by your last slide, Nelson, where it's a large number of people overflowing staircases. Because my question is about how do you engage a large enough number of people to actively contribute to your Bionetwork, which is a theme that has come up already today. Maybe for the brain network, it's more centralized, so perhaps that helps. But I wanted to ask Jose also, because he had this brilliant scheme for how to organize people. How many people are actively contributing, and how do you motivate them to spend their time on this?
It's an excellent question. I mean, I don't think that we have we figured out anything special, but I think really it was convening people together in the same location for a couple of days. And then when you help to build that trust and that familiarity, there's simply those disagreements that are okay to have in the room that are for some reason not okay to have on Zoom, and so it kind of helped people speak up, build that trust, understand that there is this divergence of opinion that's actually coming from a good place of trying to advance our understanding forward. Yeah.
And in the brain side, thankfully it's a small knit community in BICAN, so we're able to closely collaborate. But the question is, how do we reach out farther then? The previous references that we need to figure out that process.
The other thing too that I'll say is very important is even though now we've divided up tasks, at the meeting it was in such a way that everyone could be around the same table. And that meant that in original things, let's have the computational breakout session. Let's have the biological expert breakout session. No, we need to speak the same language, right? We need to learn to speak the same language and build those common, in the same way as the ontology, right? We might have disagreement in terms of legacy terms and new terms. Why is that the case, right? And so I think that we need to be open actually to more of that multilingual understanding.
And being transparent about all the links, across studies and across groups and how to share that. And hopefully CAP is a nice platform to encode all that.
It can also make it a bit more democratic if you can make it on-site for these meetings for all sort of reason. Having another platform where you can actively engage wherever you are in the world is actually really critical to our mission. Sarah, you get the last question. Oh, sorry, Aviv, I didn't see you. Well, we have our co-chairs can answer, and then Aviv.
So my question was for both of you more about how do we, or all of us, how do we start thinking about the transitions kind of from the business cells, let's say the epithelium, kind of into the lamina propria, into the muscularis, and for the brain, out to the dura, the meninges and the skull, and then the peripheral. Like, are you starting to kind of use the spatial data to sort of think in a comprehensive way to let's say phase 2 forwards?
Yeah, I mean, I think I like how you said this business end of the tissue. So the epithelial cell of the gut, I think there's obviously the microanatomical knowledge that we can really learn from these beautifully advanced, technically intricate spatial transcriptomic technologies. There's a more basic question, actually, I think that we're starting to grapple with, which is the many meters of gut that we all have inside of us, right? Or in a mouse, centimeters of gut. And the key thing is actually in terms of the nomenclature. Do we start to call things epithelial cell of the duodenum, epithelial cell of the ileum, of the colon, right? Is this useful nomenclature, or do we actually need to find consensus in the taxonomy? This is something that we actually don't have a clear answer to. And obviously the brain is littered with different regions, and I don't know how you guys are approaching this. It may be interesting to learn.
It's a complex problem because in our names we try and use the most appropriate fine region in the nomenclature as the first identifying term. So you know if you're in GPI or GPE, or if you're looking along these different epithelial or even vascular systems, like if they're unique types across different brain regions. So spatial is a nice way to understand that and encode that in the names. But also the virogenic tools give us insights to cluster level organization of neurons in the brain for just one type, which is a kind of orthogonal to spatial, but we find new patterns that we don't actually recover well on spatial sometimes.
So with that, okay, Aviv, short questions so we conclude. We're a little over time, but please say.
To answer.
Okay.
How is that? Just at least the thought is out
Food
there.
for
It's
thought.
actually relevant, I think, to the answers that we just heard. And it's really primarily a question to Jose, but I thought Nelson might have perspective from the brain. So the gut is this really, really long thing. Right? Now, in the brain, it's really big, but it's almost like every piece is different. And we know that almost up front, so they're trying to really get to completion. And I was curious, how do you get a sense of where you are in terms of what's already been done relative to the magnitude of that really, really big thing that kind of repeats itself, but is not exactly the same. And at some point becomes very, as you said, right, the jejunum is very different than the duodenum. It's not really the same. But each of them is really big.
Mm. Yeah, it's another really good point. I think that what we have to understand, and I think even more so than that, the actual, it's a little bit of that like cell subset, cell state question in a way, which is where do you draw the transitions, and are there really demarcated transitions, and how does this extend? Well, I think what we need to do is we need to really look across developmental timing, and there's really beautiful work from Sarah's group and others on this point, but also across different cultures as well, because of the noted impact of diet on that. How does diet actually influence gut maturation, and then the setup of that zonation, right? How discrete, how flexible is that? Can you go back to a different state of the gut in some way? So I think that all of these things speak to the magnitude of the problem, and when we look at all these cells, 8 million cells in the atlas, right? This is what, how many villi together in this? We're barely scratching the surface, I think, of the problem still.
It's a good point. Like we mentioned, we're only at version 1, or it's an opportunity to kind of reiterate that this is just the beginning of the journey in terms of really mapping these organs in details across space, and also to rethink about these annotations. Nelson, any last concluding thoughts?
Just on Aviv's point, I think the multi-species angle gives us a lot of power to understand what we don't know in human. Mouse's profile type we only see in the mouse whole brain atlas. And that's a nice background for us to make sure we've profiled this massive, right, human brain going from mouse very small to now massive human structures that we may not know if we've captured all the types, but mouse gives us a nice framework to understand. We've at least captured the space that the mouse has defined. And I think the additional species will give us more and more confidence that we've captured all the diversity of types across really complex brain regions.
So with that, if you have more questions, Nelson and Jose are there for the entire meeting. Please find them. Please join me in thanking them for their generosity in sharing insights about their approach to Atlas Study Annotation.
So hello everyone. So this is the next session. We've listened to update on where the atlases are, and obviously these atlases are informed by the Bionetworks. So my name is Haniffa, and I'm one of the coordinators of the developmental Bionetwork, and I'm just going to kind of like frame this session, and then you will hear from two speakers, Martijn and Malte, and then following that we'll have a panel discussion. So what I would like to emphasize is why do we need the Bionetworks, and why is the community engagement as part of the Bionetworks very important. And this is something that I really truly believe from the bottom of my heart. It really does make the knowledge of the data, what you can extract from the data, far richer, and subsequently the atlas that you build is just going to be a much better quality atlas. And also fundamentally, these atlases are for the community, so it only is right that we build something led by the community for the community. So we can't really do these atlases without the community engagement and input into it. So this is how we kind of see the way the atlases are going to be integrated in terms of having the community involvement, right from the contribution of the data sets, which gives you also a better understanding of the data sets, the limitations of the data sets, the context of which the data sets were generated. And this really requires a broad range of expertise, and we knew that was needed when we generated the data. We now know that that is equally important in terms of making sense of the data. So what we end up getting is a high quality product that will then be something that the community is really engaged with and finds it as a valuable tool. So that's what we're going to be discussing, and we'll have two speakers. I've mentioned Martijn Nawijn, who will be talking about the lung atlas and how the lung cell atlas has engaged the broader community in their efforts. And then we'll be followed up with Malte Luecken, who will be talking about adapting the integration strategies. I kind of see this, how we do the integration, as a bit of clothing, ready to wear, pret-a-porter, or kind of, high-end bespoke tailoring, the haute couture sort of side of things. And somewhere in the middle is a sweet spot that has standardized approaches that we can all use and be happy with. And that's something that we can discuss later. And then we've got a panel discussion. This is going to be for half an hour, and please engage and ask your questions. And there we will have Chen, Matthias, Kretzler, as well as Malte and Martijn. And I will try and kind of facilitate those discussions. Just very briefly, because we've seen this slide, we've got a fantastic Bionetwork to assemble the atlas from the ground up. We've got an amazing number of researchers who are engaged in each of these, and we're on the path to generating these atlases, some of which we have a version 1 of the atlas, and some of which we're still working towards a version 1. And it doesn't mean that don't be put off by the fact that there may be version 1, version 2, version 3, because that is how the Human Genome Project was. It had multiple iterations, but whatever product that was there was actually highly usable and a significant value to the community. So just to summarize with regards to the importance of engagement with the community in terms of understanding the strengths and the gaps in the data, and also understanding what we can get out of that data in assembling the atlases. And there are many strategies to compare the integration methods, and we'll need to find a space in between the ready-to-wear and the bespoke tailoring efforts that may be there. And so without further ado, I'll now invite Martijn to give his talk on the Lung Cell Atlas engagement community piece.
Thanks. Okay, thanks Moes for this great introduction. And first of all, I'd like to share how happy I am to be here. I missed Vienna due to COVID. I missed Toronto due to other obligations. So for me, it's been a while to be in this meeting. I'm Martijn Nawijn. I'm one of the coordinators of the Lung Bionetwork. There's 3 others, so it's not just me. And I was asked to tell something about how did we as a lung community or Bionetwork leads involve the broader community, because we delivered our first version of the Atlas last year, and we're actually currently very actively pursuing version 2, because if you are a little bit ahead of the curve, it also means that everything you do is not fully in line with what all the Bionetworks were supposed to do, so we had to also fall in line. And I'm going to tell you a little bit how we organize this, and I hope it's going to be useful for you. So just a little bit of a reflection on where we're coming from. Back in 2016, when we started in London with the HCA, we didn't have Bionetworks, right? This was something we invented, in a sense, along the way. And the Lung Bionetwork really got together for the first time in 2018. But at that time, there were already single-cell papers out there from data sets of lung. So this should have been a premonition for us that there's a lot of activity outside of what we're doing within the HCA. And I think that's key to understand and to involve the community, is that, you might go down this rabbit hole of building your atlas, but, out there's actually a lot of stuff happening. So we published our goals, we published our approach in respiratory journals so that we could engage with the larger community. And then, of course, a lot of data papers started coming in. And then at 2020, we were happily trying to build our first iteration of the atlas, and of course, COVID hit us. That had two problems, or it had one problem and one opportunity in a sense. The problem was that, of course, if you work on lung and you depend on fresh lung tissue, in times of COVID, this is very hard to get. So this was a bit of a problem. At the other end, it was an opportunity for us to show what the impact of an HCA, of an atlas, could be for the wider community. So we also started publishing papers as a community on, what could we already do with our existing knowledge to help understand the pathogenesis of this novel disease. And the first one of this was actually published in, I think, April of 2020, so very early on in the pandemic. And then what we learned from doing this is and this is actually this paper here, we could still call it the single-cell meta-analysis, but this is actually the groundwork of understanding how we do all the integration, how do we build an integrated atlas, and then implement this in this first integrated atlas of the lung. That was released in 2022 as a data set, but then published in 2023. So that's the history of it. So that's version 1. So are we done? Of course not, right? That's what we've heard today already a number of times. We actually need to interact with the community. So just to give credits, because that's a critical part of collaborating with your community, is giving credits to those who deserve it. Malte will be talking after me. Lisa was a PhD student, and Sasha Misharin are the people who were really instrumental for making this first version. And you can see that we worked with things like Excel sheets. And we worked with one object that everybody, so rather than having a cap, we worked with this one Celex gene object that everybody was annotating with, and then communicating through Google Drive and the sort. So this was like CAP before CAP was even invented. So of course, once you publish your atlas, you come out of this rabbit hole and you look around and the world has changed, right? So we were not alone. And there's actually, the lung is interesting because there's a large number of initiatives that are not directly or totally unrelated even to the HCA, but that are contributing really to the field in a great way. So for instance, the LungMap Consortium, which is a long-standing NIH consortium in the US, already published these cell cards, and this is something that has now been adapted in the HCA. As if it was on one of the slides of Chloe earlier today, so you might have seen it. And they actually described all the current knowledge, all the historic knowledge with a lot of evidence. And it looks like this, there's basal cells, what are the features, what are the function, what other names are there, what are the markers, where are they located, what's the experimental validation for the existence of these cells, and what's the developmental origin. And they did that for all the cells in the lung. So that's a huge resource, of course, for the community. And here is the lung map, hierarchical cell type annotation, if you wish. They were also constructing their own atlas, which was also published in 2023, just a few months after ours. And they took a different approach. So they defined seed cells and then used the seed cells and mapping all the clusters to seed cells to generate their atlas. So they went about it slightly different. They used a lot of old knowledge that was already out there to then make their atlas. Now the problem is some of their studies were also in our atlas, and we don't necessarily agree across atlases. So for lung we now have 2 atlases, one the HCA atlas, one the CellRef, which is the lung map atlas, and they don't necessarily agree. Now this is silly, right? I mean, we don't need this. Nobody needs this. So the other thing they did is they generated this web interface where you can actually interact with the data and explore, classify, query, reanalyze, etc. So they have their own sort of data environment as well, which for the lung community is really very useful. But we should also look at it and see what can we use of this and how can we make sure that we get things together. Because we don't need two sort of competing initiatives. I mean, that's just silly. So when we looked at our version, the HCA version of the lung cell atlas, we needed more variation in ancestry, we needed finer grained annotations, especially in the immune and mesenchymal compartments, more data sets from disease, we need consensus, and on the cell type annotations and ontologies, especially with the lung map, because we did not necessarily agree with each other, and we need to integrate the data sets to make one reference, right? We just want one reference for the lung, and then be done with these two competing references in a sense. How did we go about it? Well, first of all, we did sort of a systematic search, not really a systematic search, but sort of, to find all the data sets that we were not aware of, especially for this part, where we want to look at into the ancestry. We really tried hard to find data sets out there that had ancestries that our original atlas did not cover very well, and that was, a lot of ancestries. We then reached out to obtain those data sets, we integrate them, and then we align them with the lung map. So it sounds rather straightforward, but it was not. So LungMap already organized this meeting, which was hosted by Chen at UCSD in 2022, to basically discuss already existing differences of view on cell type identities and cell type annotations. And we talked to Chen, and we said, like, why don't a few of us go to that meeting as well? And we just talk to each other. We start talking to each other, because that's the most important part, right? You really need to interact with all the other people in the community out there. So Pascal Barbry, for instance, one of the other coordinators of the lung Bionetwork, he went to this meeting. I couldn't make it, unfortunately. I tried to connect online, but that never really works for a real face-to-face discussion. But Pascal went, and a few others of the HCA community went, and then we already had very good discussions and started to integrate and at least talk to each other. Then we involved the larger respiratory community, so at the ATS, which is the American Thoracic Society, so the community for the respiratory folks. The sections of genetics and genomics organized this meeting, and this was chaired by Gloria and myself. Then Malte and Richard were, who are both here today, were presenting, and we already started talking and discussing and involving the larger community. How can you contribute? What are the discussions? What are the open items that we need to solve? And how do we interact? And then we actually got this project funded, which is called the Collaborative Cross-Consortium and Continent Lung Cell Nomenclature Project. Well, this is Gloria's name, but actually what it means is that across consortia, so LungMap and HCA, we really tried to get together, and we had a workshop. So we organized a workshop at the ATS meeting in May this year, where we started discussing all of this, and also discuss what is the way forward, what's the process going to be, and what sort of decisions do we need to make, and how do we then come further. So it's LungMap, HCA is in here, HubMap is in here, and even non-consortium partners. We were able to find a few people that were not part of any of these consortia. Of course, I tried to get them into the HCA, but whatever. This is just to show that if you look at the same phenomenon from different angles, you're basically studying the same thing, and you should also use the same language. That was the point of this this slide. Okay, so what are we going to do? Well, we'll just finish this integration, so that's something that Raphael and Martijn are working hard on the supervision of Malte. Then load it onto CAP, and a huge handout to the CAP team. They did a magnificent job using Select Gene's object and metadata, so also the Select Gene team. I mean, we wouldn't be able to do this sort of activities without them. Stage released for unpublished datasets, because there's a number of unpublished datasets in there. We'll organize this annotation jamboree on CAP with these groups that came out of that meeting in San Diego that LungMap organized. So we're really trying to bring together these different community aspects of this work and make sure that all the folks that are the experts on a specific lineage sit in one of these working groups, and then we'll do the consensus annotation on CAP whenever we're ready. I mean, we promised to be ready already, but we failed our own deadlines, of course. Then once everything is done, all of this is also recorded. It's metadata, the tier 3 metadata that goes into the HCA, right? But of course, LungMap has their own system where they also want to record this, and that's fine, right? That's perfectly fine, because they also have this knowledge base. They also have deliverables that they need to meet, etc. So we accommodate all of that. And of course, HLCA version 2 will be released both by LungMap and by the HCA, as a shared activity. And I think that's a real win. If you can involve all the other communities or activities within the community out there, you can really make a difference. So, lessons learned, how did we do it? I think, first of all, you need to realize you're not alone. I mean, once you start building this atlas, you go down this rabbit hole. But you need to at some point, you stick your head out and see like what's happening around me. So you're not alone, and there's a large community doing very good science. Get them involved, right? I mean, that's the tricky part. Be proactive, reach out, be very communicative, keep sending mails, keep talking to people at conferences, and do all that. Reach out, engage, ingest, give credit. You have to give credit to everybody, because otherwise nobody's gonna collaborate. And you need to do this. It's important. Everybody needs their papers. Everybody needs their name on shared papers, etc. And you need to allow this. And also, celebrate their successes with them and support it. Evolve, update, give credit again, align, communicate, give credit again. Right? I mean, give credit. This is the tricky part, but it is very important. Actively support, everybody getting their own studies published, and help them, involve all data contributors, and give appropriate credits. Well, I've said that now. So with that, credits to whoever contributed to the HLCA, credits to the LungMap group as well, because we are on a shared journey here, and it's not us versus them, it's all of us together. And with that, I'd like to hand over to Malte.
Perfect. I'm just going to see. Okay. Back a couple slides. First slide. There we go. In the HCA integration team, we are working with 18 biological networks with up to 3,500 stakeholders, as we heard, to deliver integrated reference atlases within a 3-year time period. That is a challenging task at best, and it requires efficient reuse of methods and of expertise, while also being able to respond to the requirements of individual tissue questions. You wouldn't reuse the same dissociation protocol across tissues, so why reuse exactly the same analysis pipeline? I think in the past 2 years, we've found quite a good balance for how to strike that, where we can strike between the specificity and the generalizability across these projects. But who is we? I can just highlight a few of the people. This is really a massive effort with pictures who are actually coming to the meetings, weekly meetings regularly, but there's a lot more weekly meetings involving just, a lot of the people on this list and more. There's a few bullet points I couldn't, unfortunately, kind of write out given space. And together, kind of in a hive mind, we've tried to come up with a process that Chloe's already introduced quite nicely, that we can follow to kind of be able to deliver some of these projects in a reasonable time frame. Now, this process, having a process like this, is crucial to involve the wider ecosystem, who are enabling us to deliver that. And the people in those groups I haven't even added to the slide before, who are also critically involved, to be able to kind of really importantly kind of deliver the metadata, the data ingestion, and then some of the kind of comparisons via CAP, for example, across different cell type annotations. And they require us to have some sort of reproducibility so that they know exactly where they fit in. And different projects are at different stages across this timeline, and after this meeting, we'll be kicking off a couple of new starts to kind of move across that process. But having one process does not mean having one strategy. In fact, how questions are answered differently across different projects at earlier stages in this process determines knock-on effects that change the integration strategy moving forward. Specifically, in this kind of Atlas scope definition, or ingesting datasets, what datasets are available, different questions can come up around realignment, is it possible or not? How do we integrate cells and nuclei, or how do we learn from earlier versions? And that then has knock-on effects that I'm going to look to get into detail a little bit, just showcasing a few of the decisions that have been made differently across different projects to help you get an idea what that means. For example, in kidney, there was an effort before this integration project started in 2022, which is a CZI-funded jamboree showing where we contributed as well and found that integrating kidney datasets is particularly hard. And so the team initially set out, let's remap all these datasets to reduce as much of the potential technical batch effects that we can. However, in remapping, you can lose annotations on the cell type level because the annotations you have do not come with the barcodes necessarily. And so now we have this remapped object with different barcodes, different cells, and how do you map that to? The team took this as a strength instead, and when they were preparing to compare integration methods, they had to kind of re-annotate everything together with the Bionetwork champions in this process of extracting gene markers from individual studies, re-annotating them, taking them together to predict for the other studies, and then kind of improving this from a core data set to a harmonized effort. Now, this happened before CAP was nicely available to make this all more streamlined, so now you can do this on CAP, thankfully. But in the end, what the team gets out is a set of kind of harmonized labels at some resolution that has been kind of done together with the Bionetwork, who generated several of these data sets that can be used to compare different integration strategies. In the Retina project we've heard about a couple times already today as well, there was a similar re-annotation effort, only this time it was like, instead of going into detail on every individual study again, they performed kind of an automated label projection at a course level, taking into account that maybe these labels are not as high quality as could be, but they're still useful. Specifically useful to understand how to guide method selection moving forward, but maybe not sufficient to supervise this integration process because the trust in the labels that are predicted are not necessarily as high. This has knock-on effects again for the strategy. One of the challenges in the Retina integration is the separation between single cells and nuclei in the data sets. And this was found quite early on as a major source of batch effect. And so the team looked at where this has been done successfully in existing studies or in benchmarks and tested these different approaches in a kind of a benchmarking setup to find that the top performers and several of these good performers actually relied on cell type labels, which was decided was not a sufficient thing to do in this setup. And so instead, comparing the individual integrations of cells and nuclei to the combined integration, the decision was made to build two separate integrations and annotate these in tandem, and you've already heard about the annotation efforts together with CAP for the Retina. In the gut, as we heard from Jose, the question of integrating cells and nuclei arose as well. The target of having landmark studies included meant that one of these data sets, 700,000 cells, was very important to keep in the reference, which is a HubMap publication, but turns out to be challenging to integrate. After kind of getting consistent sets of course labels and benchmarking different methods, the problem still had not been solved of integrating cells and nuclei. And so what the team did, they went back to earlier decisions and looked at different choices of batch covariates, studies or donors, how to parameterize the methods, and how to select features that maybe improves this integration. And specifically, they found that unsurprisingly, maybe mitochondrial genes, ribosomal genes were not as present in single cells, single nucleus than single cells, but in turn, there are more genes profiled in single nuclear data, likely because the amount of reads are distributed now across a larger number of genes. And that led them to kind of their current integration that they're working off of, which has custom kind of parameterization and feature selection to improve this integration. And they are now working together with CAP to already annotate lineages where the integration is of sufficient quality to move forward. The projects I just shown you all have the kind of the goal to understand cell type variation as a version 1 effectively. In the lung, as Martijn already mentioned, we have the kind of the luxury of learning from that version 1 with positive but also more critical feedback from the community, in some cases criticizing the diversity quite immediately upon initial publication, which is very well taken, and also something that I think Chloe's criticism of immune cell resolution is something that came up before. We clearly can do better than 3 T cell subtypes. And so one of the strategies the team used to try to tackle these limitations is build a dedicated kind of seed annotation pipeline to generate higher quality immune or higher resolution immune cells annotations, taking a subset of the data sets which have more higher resolution annotations, building with protein data, building kind of an integration of these and then projecting this to new data sets. And first, this seems to be working to a certain extent. And then these labels, these higher resolution labels, will be used to compare integration strategies to make sure that the integration strategy we now use is better suited to highlight variation on this more higher resolution immune cell populations, and thereby kind of learning. And a last use case, the immune integration came up with several specific questions in the Atlas scope, looking at inter-individual variation to large scales, lineage-specific integration to really kind of pull apart those details per lineage. And they also, in this project, we also did partial remapping, but only for the data sets where we have the matching barcodes and cell type annotations so that we preserve the cell type information. After generating kind of consistent coarse lineage labels to be able to focus on a lineage per lineage level, there's specific feature selection, specific QC, and integration parameters chosen, leading to kind of a first preliminary integration, in this case showing that actually there are still issues that led to kind of an improvement, iterative optimization of the feature selection process. And that then will go towards a preliminary annotation of that integration after kind of optimizing this feature selection. And so, as you see in this way, the different ways of kind of combining information or using the same process can lead to very different strategies. Crucial in this is the continuous feedback between the Bionetwork and the integration team. And that is a challenge at 9 million cells. It's a challenge at 1 million cells. It's a specific challenge at 9 million cells. So what Michaela from this team wants to do is try to say, can we take this process and power it with a reproducible toolbox that gives all the integration team projects kind of a consistent set of framework in which to test tools that have been used in other projects, but also to kind of reuse these methods if they seem to have worked. And so the aspects of that process that are kind of more technical rather than kind of scope defining all now have individual methods that are associated with this, and we just kind of opened this up in the interest of making this a kind of a public development as well. This is still very much under active development. It's been used by several projects, and this is specifically optimized for reproducible stability, but also scalability to large scales while enabling customization, customized configurations. So with that, I hope to convince you that actually having this consistent process means very different things for very different projects. Whether that's on remapping, generating consistent labeling for comparing integration methods or cell-to-nuclear integration. And I need to thank and highlight also several posters that if you want to take a look and chat with the relevant people. And I'm going to very quickly use this opportunity to mention that we are hiring a small lab. So if anyone's interested, please let me know. Thank you.
So thank you very much, Malte and Martijn, for the insights that you've experienced. And I'd now like to invite Matthias Kretzler and Chen to join the panel. Please come up. Here's your microphone between the two of you, where I think we will focus the discussion on the sort of experiences from the representative biological networks. And I'll start off with a question for each of you to say 2 things of the sort of like lessons learned from your experience of doing the integration. So perhaps Rui, you would like to start first.
Yeah, so thanks for the opportunity. For the eye, we are very small organ, so it's much easier to guts and brain, but we're also unique where there's a lot of different parts, right? So the cornea is quite different, the retina. So I think one of the challenging is really reach out to the community, which I'm not familiar with. I'm in retina, so it's easy to get people involved in retina, much harder to get people to involve in the anterior surface. I think that's lesson learned is you need to find champion for different parts, and then they can self-organize their community. So I think that's the major thing. Yeah.
So that would be your major take-home in terms of like getting assembling the community that's going to be engaged and captured to sort of like deliver the integrated atlas.
Yes.
Okay. Matthias from the Kidney Bionetwork.
Yeah, I think outreach, effective integration of the different data types, of different procurement setups, so that we can learn from the strengths and weaknesses of each of the data sets. And then, as we discussed in Toronto, metadata, metadata, metadata. Your data only is as good as the information you get, and that obviously is the hard part. But clearly what Malte introduced us, having a robust framework available where that can be easily and effectively populated when we reach out to stakeholders has been a big step forward.
Fantastic. Malte, you've given your talk, but you've got your microphone. So
so I think because metadata and early engagement was already said, I think for me the two parts would be, especially across this scale, is really thinking about whether each individual step, some of these steps are very time consuming, like metadata correction, remapping, the value of that step versus the kind of the cost, right? And considering that and how needed it is, and to perform kind of small scale tests of whether this is really needed. And the second aspect, I think, is the value of having this kind of reproducible pipeline, because feedback happens at all levels, all the time, and you need to be able to react to it fairly quickly. And so having a consistent approach, especially to enable this across many different projects, is important.
Brilliant. And Martijn?
I think for us it was getting this multidisciplinary team together, because you need biologists from all expertises. I mean, we started out with mainly epithelial cell biologists in a sense, so we were ignoring 80% of the lung. So you need to have all that diversity in biology, but then you need a strong computational. So we had Malte, so we were good. But other than that, you need all this diversity there. And then I'd like to mirror something that Aviv said earlier today. You also need to really consider, what is good enough for version 1, and stop, and then get it out, because that will generate a whole lot of feedback to actually improve for version 2, because there's going to be a version 2. There's even going to be a version 3 and 4 probably at some point. Maybe not with me, but someone. But yeah, I think that's very important. Like it needs to be good enough, but it also don't try to make it perfect, because you'll never end, and then you'll also never get all this feedback.
So that's fantastic. So community is very important, and perfection is the enemy of good, and metadata, metadata, metadata. That seems to be the ringing endorsement here. I'm now going to ask the question a slightly different way. What would you not do again? So maybe we can start with you, Matthias.
Yeah, I think what we just discussed, let's say when we are good enough, and when we call it, yes, we are waiting for people to respond, we are reminding.
So is that reflecting on the kidney journey in terms of like, you feel that maybe you've done, you're trying to achieve perfection? Is that the kind of lesson that you shouldn't have done that?
It is, and obviously the field is moving so rapidly. Having single cell and single nuke data consensus annotated is a foundation, but everybody looking at the sky and not is looking how to build the foundation. So that's where this is a time window we have to move. We have to move quickly now to retain engagement in that. And CAP, I think it's really, and Ivan and team, congratulations that you pushed so hard, will be a critical element to say, look, we go with relatively low resolution where we can build consensus. For every else, we will have a synonym structure which will be good enough for that first version to come out.
Okay. So what would you not do again? So what mistake or what lesson that was learned that, it's just for the rest of us. It would be quite helpful to know what not to do.
Yeah, I think, so it's really hypothesis, so I cannot go back time and do it again. So I think what I would not to do is, like, I simply send an email or go to our meeting, we have annual meeting, and say, hey, here's the atlas, who want to volunteer? It seems like very hard to get people to volunteer. So I think it's really actively go after people you think will be major leader for the different parts and then let them organize. So I think just be more proactive and then identify a couple people to start with. Yeah.
No, that's brilliant. So that's a good thing to learn. And what about Martijn and Malte?
Yeah, I agree. I mean, we've sent out quite a few emails early on through the Lung Bionetwork email address, in general asking for input or data sets, and you don't get a whole lot of useful. I mean, of course, some data sets we acquired that way, which are very useful, but the more proactive going out, finding data sets that address the shortcomings of your atlas that you're aware of. In our case, which ancestries was really a big thing. We really want to chase that and then be proactive, which takes more time. So that also means the planning of your process. Our lung atlas sort of organically grew initially, which is not the way I think to do it. You should really make like a project management planning. Really say, okay, in 2 years I want to be there. How do I get there from where I'm now? And then really start that process and dedicate time. So that also means you need to dedicate time.
So it's like Jose said earlier about having a training plan. So we need some sort of like target. Yeah. And Malte, because you've handled several of these, atlases.
Yeah, I think
what wouldn't you do again? And how to mitigate
against it? I think we're learning quite quickly from this. So I think one of the things that we kind of sometimes notice early on is that this kind of the second point on the process, atlas scope definition, is something that you shouldn't undervalue. People have different expectations of an atlas, and it's challenging if that comes out later when you're evaluating integrations, rather than in the very beginning when you're understanding how to target which data sets you should include or not. And the more specific you can be on the exact expectations everyone has, the stakeholders has, always what an atlas should do, the better you can encode them in how you will compare integration methods to be able to deliver that. So I think like just starting without knowing exactly what the target is would be the thing I would.
So I'm going to now, thanks very much for all of those thoughts. They're really helpful. So I'm going to go back to kind of like one of the key things, which was the importance of engagement. Simple emails not going to be, helpful, but actually proactively looking for the right individuals. Are there things that we haven't quite done that we could do to actually enhance that community engagement? Who would like to go first?
I think one aspect is, particularly if you are procuring samples from living individuals, and this is something we have developed in the kidney-specific activity in KPMP, that we really reach prospectively and effectively out to participating individuals and have communication ways out there. And we are very proud that our first sharing tools was for participants to access their own information and samples. And that's something I consistently miss in this audience. I'm a kidney doctor myself, so I'm close to patients with kidney problem. And I really would encourage you to have that vision also in place. Not only we reach out to scientists, but do we reach really out to the people in the end we want to help and provide them path forward in their disease journey in the end. That obviously is irrelevant if you're working with diseased sample material. If you don't, I really would recommend that you do that.
Yeah, no, that's a good point, actually. I mean, it kind of touches on what in the UK is kind of called public engagement and patient involvement, which is becoming more central to a lot of projects that involve clinical samples. So perhaps that's something that we should be thinking about. I remember HCA, one of the things we did in the UK was a community engagement effort, and there we actually had an art science exhibition and worked on how people felt about organ donation and how their data was used, and there was quite a lot of valuable lessons from there, including the vast majority of people, by and large, were really, really happy that their data was going to benefit the broader humankind and not necessarily where that data was going to be of personal benefit. So I think perhaps more targeted what is labeled affectionately as PPIE activities will be helpful.
And it also is relevant for this audience in the room, why we are here, why we are doing it, and if you hear that as a scientist directly from somebody affected where your activity will change, That's also a great motivation for the research teams at large. And most of all, sometimes if you have a patient panel, some of those patients don't come back, and then the other patients will remind you, do you think it's really adequate that you did discuss authorship for an hour when 2 of the 12 of us are not in the room any longer? And that also helps to ground us.
No, that's brilliant. Any other thoughts? Rui and Malte, I think you...
Yeah, so I think one thing during the process, we realized the need of community is quite different. So it's very diverse. So I was actually personally surprised how many people email us, ask for Excel file. So here's the Wiley, why not just go there? But there's so many people are different. I think it's important for us to give the data back in form and shape that community really can consume a benefit early, even before publication, so that you can understand their need, because they email you, ask for things, and you can adjust what you need to present. And also thanks for all the the software available. I think it would be nice. We're still working on that in some way, so you can put all those different tools together, people can navigate easily. I think still pretty overwhelming to look at all those tools possible to get data from.
I think that's a really good point, because it talks about not just engaging while we're doing the integration atlas, but actually continued beyond once that atlas is published. And it comes to a topic that's quite close to my heart about having a web portal that people can benefit in the same way that Ensemble, revolutionized the data from the Human Genome Project. And maybe this is that continued engagement that happens not just during the assembly of the integration of the atlas, but actually well beyond that. So that's actually a really valuable point. Thank you, Rui. And Malte?
I think we've been thinking, data contributors are crucial to this, right? And we've been thinking quite long and hard of how to engage as many data contributors as possible. I think an area where we haven't been thinking as much how to engage, there is engagement in some cases as computational biologists. So I think we often see that after an atlas comes out, other use cases of these atlases appear as they've been like after publication. But actually there are computational people within these bionetworks that could be empowered to address particular questions they have in a computational way once we get to a foundation. And that has happened to a certain extent. So in Martijn's lab, Tessa is around. She kind of jumped on HLCAV1 and did deconvolution, right? After seeing, oh, this is something I could do. And so I think ways to reach out to the computational bionetwork community, I guess, empowering specific tasks, because building this, we don't know all the things that could be done with it.
Yeah, no, I mean, I think that's another great point. And if I kind of look back at the clothing analogy, it's like taking the ready to wear and altering it so that it's better fitted to the body and shape of the data set, so to speak.
Yeah, this example that Malte gave was really driven by the community, because we were getting so many requests for people, can you help us deconvolute our bulk data based on your atlas in those consensus labels, which of course makes a lot of sense, but we never developed that. So, and now Tessa did. So that's great. Yeah. The other thing I think that where we missed the mark a little bit early on in HCA is this very strong focus on healthy only. I think because a lot of the money and therefore a lot of the project sits in disease areas, also a lot of the use cases sit in disease areas. And I'm very happy to see now that for the spatial aspect, we're actually going to do this. Because it's not just where all the data sit and all the money is, and therefore it's easier to engage with community because they are already funded, which is not trivial. It's also the fact that a lot of your critical cell states, if you wish, will be revealed in a disease context, and contrasting that to the healthy that everybody knows.
No, I mean, that's a fantastic point. And I'm going to open it to everyone here if you have any questions relating to it, and kind of casting the net a bit broader, because you've had the kind of lung, multiple atlases, the kidney and the retina, and I'm going to come from my involvement with the skin, where we've taken a slightly different approach. We've actually gone by annotating by lineage as a start, and then using that data and leveraging the cell annotation platform, which I keep calling community annotation platform, and I quite like that name, as a way of having that lineage kind of, locked in, and then other, groups working on the different lineages, and then assembling it. So I guess there are kind of many ways to achieving the integrated atlas, and it's really amazing that cell community annotation platform has been extremely flexible in supporting, that sort of use of that platform. So I think, Sarah, you have
Yeah,
have a
no,
mic,
I just
and then
wanted
there's another mic
to
there. Sorry.
pick up on what Martijn said about the disease, right? Because that's 200% true. Is a lot of funding comes through the diseases. Tumors, a classic example. Our funding for the lung first came from asthma and industry funding. So, but I would turn that on its head and say, yeah, but we mustn't forget the healthy, because that allows us to interpret what's changing in disease. So Human Cell Atlas is relating to all the diseases which are relevant for the individual tissues, but that interpretation is enabled through the healthy reference. So but of course we work in partnership, with all the disease programs. I mean, not a shadow of doubt, not for 1 minute is that a question. And it's a constant partnership and a constant dialogue.
Yeah,
no, but that's a good point. But
Matthias is a clinician.
Sorry, yeah.
And the point is exactly that. And mapping the continuum from health in disease. And the beauty is that's what these tools do. I've done 20 years of research on bulk, and it was difficult. And now, as we can anchor, we find our healthy states, our healthy neighborhood in heavily diseased samples, and conversely, you and my kidney for sure has areas who are as diseased as our patients headed for dialysis. And that's an incredible lesson to learn. It comes with age, it comes with socioeconomic implications, it becomes with genetic backgrounds. So that's a strength. The final piece is with using disease samples in many organ systems, we routinely obtain biopsy where you are under full control of the material, you don't have any procurement ischemia, none of the other confounders, which disease material always limit what we can learn from.
So, Aviv, is it relating to the same point?
Yeah.
Okay.
Two really briefly. First, just last one to what Matthias said, that the healthy, all of us carry plenty of things that we think of as disease. It's not really that categorical. But second, that one of the things that becomes really dramatically shifted in disease is just compositions. And cells, which are quote-unquote healthy cells, but extraordinarily rare in the healthy state, can expand in the disease state and be a lot easier to detect. And then once you know they exist, fishing the handful of them from the healthy tissue becomes a lot easier. There's actually a nice example of that with tuft cells in the lung. Yeah. And there's others like that. So the dichotomy, if our mission were to assemble all the diseases as well, we would never finish, et cetera, but we should be pragmatic about it, because there's so much there and we need it.
Yeah, I agree. Sorry,
Yeah, I wanted to follow up on Malte's last comment about engaging the computational community earlier on. I think it would be really valuable once you have that integrated data set to release that to the computational community, because I think they have a lot of interesting things that could be contributing to version 1 Atlas. Not version 2 Atlas, but version 1. And so if there's a way to do that, freeze down that data set, distribute it, and then see what comes back, see what interesting things come back from the community.
So that does happen to a certain extent within the Bionetworks.
Yeah, so. Sounds an excellent suggestion. We have another question.
Hi, I have a question for Malte. Just a follow-up to the same question, and Malte, you touched upon the scoping of atlases. So coming from a consumer's perspective of these atlases, and choice of the technology stack, choice of integration methods, of course there are metrics to quantify for how well your mixing of batches is, and then how well the recovery of biological information is. But talking about knock-off effects of these, or knock-on effects of these methods, sometimes it might become difficult to, by choice of, let's say if you use Harmony, then doing out-of-sample querying might become very difficult on those samples. The second aspect is also the question of IP. Some of the methods have very restrictive IP and you cannot use them for commercial purposes. And how does that feed into the choice of technology again for integration purposes?
Thank you. Malte.
So it's easier to answer the first question. Something we definitely take into account is like when I mentioned kind of encoding this kind of expectations in the scope of an atlas, if we wanted to generalize to new data sets to be able to help annotation, something that feeds into that decision is there a method that can enable us that to happen? SCArches works for deep learning-based methods based on SCI tools frameworks. Harmony has Symphony, which is maybe limited in the new cell type identification, and then Azimuth seems to be generalizable across these tools. So but there's no way, and in some cases I think I could shout out here the Skin Bionetwork, where there's a dedicated effort to develop a tool to enable this, but it's definitely a part in the decision for which methods we even test in the evaluation. So things that scale, things that enable the downstream use cases. The second question is a little bit harder. I think maybe it's a follow-up.
Yeah, okay. So that's like a for longer conversation. But in any case, I think I'm going to bring the panel discussion to an end. It's been really fantastic to hear from our panel members, their experiences, what they say don't do, what they say we could do in the future, including some suggestions of how disease may help, and new ideas for PPIE and web portals. So really exciting times ahead for the Integration Atlas and beyond. And with that, I'd like to thank everyone for participating, and thank you, Martijn, Malte, Matthias, and Rui, and we look forward to Piero's final remarks after this.
So I will be very short because I don't think we need a very long remark, but I would say that we understand how important it is to collaborate, and I really hope that you will reach out and try to understand. You'll use breaks, lunch breaks, and coffee breaks to really have the conversation. So just I would like to mention we have, it is not a typical Sunday Italian lunch time. It's going to be shorter. And we have only 40 minutes. But please come back at 1:50 for the technology showcase from 10x Genomics, Oxford Nanopore Technologies, Illumina, Element Bioscience, and the Viz. And for the raffle to 50 to